Karan ma'am, start.
So landing slide देते ना.
Okay, so good afternoon, everyone. I welcome you all to the 3-day long Foxy ICOG online certificate course on genetics and fetal medicine. To begin this academic feast, I would like to welcome today's convener, Dr. Sita Ramamurthy Pal, ma'am. Ma'am is a consultant fetal medicine and obstetrician at Apollo Multi-Speciality Hospital, Ankura IVF, and Fetal Medicine Center and Institute of Fetal Medicine, Calcutta.
Thank you.
And
Thank you. That's enough. That's enough.
Okay.
Thank you. So welcome one and all. Before we start the program, we'll invoke the blessings of Lord Saraswati and Ganeshji as well. So can you please start the song for the inauguration? Thank you, Nilima. So welcome one and all to this yet another Foxy ICOG certification course on genetics and fetal medicine. We've had one in 2023, which was a huge hit and was very, very popular. And from that, people have been asking that we should have one more of this sort. And thanks to Dr. Parag Beniwale and Dr. Sarita, we've been able to do that. And it is with I thank all the faculty who've, agreed to take part in this and participate and share their knowledge. And especially Dr. Ratnapuri man, who also helped in formulating the talks and what would be best for our obstetricians and the postgraduates and the geneticists as well. So thank you once again. And without wasting time, I would like to formally inaugurate and introduce our president, Dr. Sunita Tandulwarkar. She's busy in another meeting, so she will not be able to join. But as a formal introduction, she's our current visionary dynamic president of Foxy. Amongst all her credentials, academic credentials, I think the most important thing she has done this year within 2 to 3 months of becoming the president is her multitude of projects for the Committee for the Women of India, her Sampurna project, her Do Teeke Zindagi Ke, which has become viral all over the country. So thank you, madam, and with her Gyan Pravah podcast series, which is going on very well. So thank you for all these initiatives, and thank you for letting us go ahead with the academics in this part as well. Next, we have our very own, my very good friend, of course, Dr. Parag Beniwale, who is the chairperson of the ICOG now. And again, a big thank you to him for giving us this opportunity to convene and have this wonderful academics of genetics and fetal medicine, especially when genetics is so very vital in our day-to-day practice, be it obstetrics, be it gynae, or be it any part of medicine from head to toe. We can't forget genetics. So that is where we are. So thank you, Dr. Parag, again for this and for giving us this opportunity. Next, I would like to thank Dr.
Sita, why don't you ask Dr. Parag to speak, then
Yeah.
Sunita is joining.
Yeah, okay. Dr. Parag, can we have a few words of wisdom?
Thank you, Sita. Good afternoon. I bring greetings from ICOG, and it's indeed a pleasure to see you all again with yet another certificate course from ICOG. And I must appreciate efforts put in by the 3 conveners, Dr. Chinmay Rath, Dr. Sita Ramamurthy Pal, and Dr. Pooja Loda, ably assisted by Dr. Kanan Kotadawala, Dr. Navya, and Dr. Aarti Yevle. A big thank you to you, because this is something which we had attended last year, which was a big hit. And we thought that we should have a repeat performance, because fetal medicine and even genetics has changed the current obstetric practice. And with so many fetal medicine experts, we are offering much better care to our pregnant women, especially in high-risk pregnancies. And we are able to diagnose so many things much earlier. So the program this time today we'll be having the basics of genetics today, which will be dealt with by who's who of geneticists in the country. Tomorrow we'll have all about fetal medicine, and all our fetal medicine experts will deliberate on various aspects. And of course, the third day will be very different because we'll be focusing on the genetic aspect of gynecology. So I am looking forward to a great learning experience, and there are so many stalwarts who are being part of this program. So happy learning, everyone. Please don't forget to attempt the post test, but you have to be present for all 3 days, attempt the post test, and try and secure more than 50% mark in order to give you certificate from the ICOG. My best wishes for the program and happy learning, everyone. Thank you.
Thank you, Dr. Parag, for that. And I agree it will be a great academic learning. But as Dr. Parag said, a slight change from the last time. We don't have a pre-test here. We will listen to all the talks, and then at the end we'll have a post test for which the link will be posted, and then you can attempt. But for that you have to be present for all the lectures to attend to answer those questions. Thank you once again for this, Dr. Parag. Thank you.
Sita Sunita has
Sunita is
joined, so let's request
Oh,
her
okay,
for her
okay,
address.
okay. Thank you, madam, for joining. And this, can we have a few words of wisdom and blessings from you before we start our sessions?
Thank you, Sita. It's always wonderful to see you all. Such an academic platform of ICOG. Dr. Parag, congratulations. You have chosen such a fantastic conveners. Sarita, fantastic. Parag and Sarita duo combination is going very well, very well. And I really congratulate for this fantastic idea and the way this program is going to go. Not only this program or any other master classes, it's amazing way the ICOG is working. And I'm here actually because ICOG was doing such an excellent academics. I had stopped the webinars of all my committee chairperson because it is too much of a bombarding. And I can see that 250 plus joined. That indicates that if you give a few and particular topics, definitely people still have an interest in joining the webinars. To give you great news, we are coming out with the physical postgraduate courses for every state. And I think Dr. Ashish Mukhopadhyay and Charmila will run that program. We are running off from 15th April, and all teachers over there ensure that you cooperate, you help, and send your students for these courses. Thank you so much once again for giving me a platform to address all of you, and it's always pleasure. Thank you.
Thank you so much for joining, Dr. Sunita, ma'am. Thank you so much. I mean, it's amazing to see that despite all your activities as well, you have found time, and plus you're also, doing academic activities along with the other social activities. So hats off to you. Before you joined, I was just briefly telling about how many projects and initiatives we have started in the last 3 months, and they are going at jet speed in all the colleges and all across the
You will all, I don't know whether you will spare me or not. Last evening, brainstorming, because we are coming out with a very huge project that run for 3 years or maybe more. Two more big projects I'm coming out with. We'll tell the time, we'll answer it. Let's not spoil the academic time for it.
Thank you so
Thank
much.
you. Thank you.
Thank you for your words and encouragement. Thank you. So next, I would like to formally introduce our Secretary General, Professor Suvarna Khadilkar, who's again slightly busy, so she may not join, but she's been a big help and support for all our activities. Dr. Ashok Kumar, who's the Director, who's the Vice Chairperson 2025 and Secretary has been the Secretary of ICOG 2021-2022, another very big academician and who supports all our activities. And of course, our very own our Secretary of ICOG, Dr. Sarita Balarao, who's been so cooperative and helpful, and giving us a free hand and organizing these things. So thank you a lot. Thanks a lot, Dr. Sarita, for this, and highly appreciated from all of us, all Foxians across, to both the chairperson and the secretary. So again, as I said, that we have a 3-day course, as Dr. Parag said. The first day is on genetics, basics genetics, where the who's of who, literally the who's of who of clinical genetics have agreed to share their knowledge, and they will be giving their talks. Second day is on fetal medicine, and the third day is on genetics and gynecology. So attendance for all the 3 days is compulsory. There will be no pre-test. There will be a test, post-test at the end of all the lectures, where you will be given a link in the chat box as well as a QR code, where it will take you to a Google a box where you can attempt all the questions. But you have to attend all the 3 days and give the test and pass with 50% marks on each day. Only then you will get a certificate for this from the ICOG. Certificates will be duly sent within a month. And this is basically the course certificate is an acknowledgement of attendance, right? So this is what it will be. This is the flow of events. After the post test, you can post your questions or queries in the chat box. But once the post test is over, when all the faculty will be still there, we can address those questions and queries, as you know, some of them might not be fully addressed in the post test questionnaire, because that only 20 of them will be there. But if you have any other questions that you want to ask the faculties, there will be time for discussion at the end of after the post test, right? So with that, I hope anybody doesn't have any questions, but if you have, you can please put it in the chat box, and we will start off with the academic session. So may I please, first of all, I would like to thank Dr. Parag for giving us, for the 2 conveners who are going to be joining with us, Dr. Chinmay Ratha, who needs no introduction in the field of fetal medicine and ICOG. Can we have the next CV, Nilima, Gauri? And Dr. Chinmay will be the convener for the second day, and Dr. Pooja Lodha will be the convener for the third day. And then we have our young guns, the coordinators, who will be Dr. Kannan Koddawala for today, Dr. Navya Pillai for tomorrow, and Dr. Aarti Evle on day 3. So we have the brief CV in front of you. Yeah. Can we have the CVs? Also, we would like to tell about this MRCOG course, which is going to be held in the Foxy office, Part 3 MRCOG training course in Mumbai, which is going to be conducted by ICOG, which is going to be held on the 8th and 9th of April. We have this is an official RCOG exam course with faculty from the UK and also Part 3 examiners from India who have experience in taking these Part 3 OSCE exams. So the last date has been extended to 31st of March. So please be, if you all want to register, you can go online and register. You can visit the ICOG website. You will get all the details. And for any queries, you can mail the office as well. So moving on, again, thanking the conveners again, Dr. Chinmay Ratha, Dr. Pooja Lodha, and the coordinators, Dr. Kannan Koddawala for today, Dr.
Navya for
Navya
tomorrow.
Pillai for tomorrow, and Dr. Aarti Evle for the third day. So as we said, again, I'm not going back. The first day will be on basics of genetics, clinical genetics, second day fetal medicine, and the third day of genetics and gynecology. So with that, I will hand over the academic session to our coordinator for today, Kannan. Can you please introduce the chairpersons for the first session and then take it forward? Thank you.
Yes, thank you, ma'am. Our session 1 coordinators are Dr. A.G. Radhika, ma'am. She is a senior consultant at University College of Medical Sciences and GTB Hospital, Delhi. She has special expertise in evidence-based healthcare and guideline development. And she had held various prestigious positions of Foxy, Jogi, and also for the guidelines at International Network. And she is also recipient of many prestigious awards and 75 plus publications in international and national journals. And our other chairperson today is Dr. Meena Samrat, ma'am. She is senior consultant and HOD at Department of OBGY at Kurji Holy Family Hospital, Patna, Bihar. She was past president of Patna OBGY Society. Hello. Okay. And please, ma'am, she was also vice president of ISOPUB and also many other prestigious positions and had many publications in reputed journals. So Meena ma'am and Radhika ma'am.
Thank you, Kannan, for the introductions, and also we would like to thank the organizers for having us here. Thank you for the invitation. So we go on to the very first speaker of the session, Dr. Sakshi Yadav. She is a clinical geneticist at Motherhood Chaitanya Hospital at Chandigarh and the Care Diagnostics at Zirakpur. She has, to her credit, the fellowship in advanced antenatal ultrasound at Mediscan Chennai, observership at ISHGG NIH Bethesda, USA, multiple publications to her credit, and she is FMF certified for NTNB scan and also for anomaly scans. And her areas of special interest include reproductive genetics, prenatal diagnosis, fetal autopsy, genetic counseling, molecular genetics, disc morphology, diagnosis and management of rare disorders, oncogenetics, and hereditary cancer. So that's a huge, what would I say, almost the entire length and breadth of this earth. So warm welcome to you, Sakshi, and over to you.
Good evening, ma'am. Thank you so much for this kind introduction. And first of all, I would like to thank Foxy and Sita ma'am for giving me this opportunity. Now, without wasting time, I would like to start, ma'am. I'll share my screen. Just let me know if... Ma'am, is my screen visible?
Your screen is visible, but the slides.
Yeah, but
Can we enlarge? Is it
no,
possible for you to? I think Sakshi, if you were to use that would be better.
This is better, I think, ma'am.
Yes.
it's better.
Let's start. So, good evening, everyone. So, the outline of today's presentation that will include the basic structure of genetic material, different types of variations in the chromosomes, as well as DNA and genes, and finally the modes of inheritance. Now, since earlier we used to have only one test, which was karyotype, but in this era of genomics, there is sudden bombardment of these tests, which include FISH, CMA, clinical exome, whole exome, and finally whole genome. But I think it is very important, before we know about these tests, it's very important to know about the basics, basics of genetics, which we are going to discuss today. So this genetics is basically, it includes your biochemical genetics, which deals with the inborn of metabolism, enzyme assays, protein functions. Then we have cytogenetics, which deals with the chromosomes, molecular genetics, which deals with the DNA. And then we have this clinical genetics part, which deals with the clinical aspect of these genetic disorders. And now even clinical genetics has broader aspects, which include cancer genetics, reproductive genetics, we have metabolic genetics and immunogenetics. So let's come to the basic part, which includes, we know in a cell there's a nucleus, and this nucleus basically contains all the hereditary material. Grossly, if we speak about, there are these chromosomes. When we open up these chromosomes, they are thread-like structures which form the DNA. And when we go much deeper into this is just the arrangement of these bases, which is basically purine and pyrimidine. So if I compare it to a library, in the library, when there are stocks of books, they are the chromosomes. When we open one book, it has these indices, which is in the form of, which can be compared to the genes. And when we open one chapter and see inside, you can see these are the small alphabets which are written in the form of a sentence, and they make a sense. Similarly, in the gene, there are these bases which have a specific arrangement of these bases, and they finally form a functional aspect of gene, that is the protein. So this is the overview from chromosomes to DNA. So we have in total 46 chromosomes. So this on the left side, you can see, this is how these chromosomes look under the microscope. So they are the thread-like structures. In these chromosomes, you can see there are alternate dark and light bands. And this is basically done by Jimsa staining. These light bands are euchromatin, which contains the functional genes, and dark bands are the heterochromatins, which contains the non-functional genes. So when we arrange this, you can see these chromosomes are staggered around when we see them in the microscope. But when we arrange them in a systematic order, in accordance with the size, you can see this is the largest and these are the smallest. Depending on, so this basically is known as karyotype. Okay, so this karyotype gives us an overview of all the chromosomes, their numbers, and if there is any structural rearrangement which occurs in a chromosome. Next, we move on to the genes. Genes are the functional aspect of DNA. It is very important to know what is an allele. So as we know, we have two sets of chromosomes. Each chromosome is present in two numbers. That means each gene has to be present in two copies. So these are the two alleles of a gene. So they can be similar, then we call it as homozygous, or it may be possible one allele is different from the other. We use the term heterozygous. So these terms I am using because in rest of your sessions, you will be hearing these terms again and again. One term is genotype. Genotype is the genetic constituent of an individual. Whereas phenotype tells you the physical characteristics observed in an individual. Apart from this nuclear DNA, we have mitochondrial DNA also. The importance of this mitochondrial DNA is, though it is present in every cell, but as we know, when the embryo is forming, the mitochondrial part is basically transferred from the ovum. So if there is any mitochondrial disorder, that will be transmitted to the mother to all her children. So this is a typical mitochondrial mode of inheritance, which is different from the normal Mendelian modes of inheritance. Now there are around 3 billion base pairs, and it will be surprised to know that only 1% of our DNA is coding DNA. The rest is non-coding. That codes for introns, various regulatory elements, centromeres, telomeres, repetitive sequences, and it doesn't mean that is not functional. They are functional. To some extent, we have deciphered the functional aspect of these repetitive sequences, regulatory elements which control the functional expression of a gene. And roughly there are 20,000 genes, but the number is continuously increasing as the research is going on. Then you will hear the term mutation and variant. So initially mutation means any change in the primary nucleotide sequence, which is rare. Frequency is less than 1%. But nowadays we don't use the term, rarely use the term mutation because that used to have a negative impact. So nowadays we use the term variant. Variant which may be disease causing or variant which may be a benign variant. Now there could be somatic mutations, mutations which occur in the particular organ, in particular somatic cells like cancer cells, and they are not inheritable. Then there could be gonadal mutation, mutations which are occurring in the germline cells, and they can be passed on to the next generation. Next term you will be hearing is copy number variant. What is this copy number variant? So as we know, each chromosome, each segment of DNA is in 2 copies. Okay. But when there is either 1 copy or 3 copies, that is deletion or duplication, we use the term copy number variant. Okay. So let's move ahead to look at the variants. Variants or changes that can affect either the whole genome, chromosome, or the DNA. So any guesses where the variations can occur throughout involving the whole genome? So the example is triploidy. Now you can see in this triploidy, the whole set, instead of diploid, it is now triploid. So there are, instead of 46 chromosomes, there are 69 chromosomes. So whole genome has been affected. So this is an example of triploidy. Now this triploidy can be maternal or paternal. So briefly, I'll discuss paternal, generally present as partial mole with increase in beta HCG and other complications. Maternal triploidy mostly present as severe asymmetrical FGR with decrease in the levels of beta HCG. So whenever we suspect triploidies, we always do invasive testing to confirm. Variants can now affect at the chromosome level also. Okay, so if I go to the chromosome, there could be numerical abnormalities. Everybody knows trisomy means 1 pair of chromosome instead of 2, it is 3. A very well-known example is trisomy 21. Monosomy, mostly monosomies don't survive. Monosomy X is one of the chromosomal abnormalities which can give to live birth. Then there are marker chromosomes, extra chromosome, which does not have any origin or we don't know the origin. And then there are sex chromosomal abnormalities like Klinefelter syndrome, triple X syndrome. So they are basically that affect the number of chromosomes in a cell. Then chromosome can have structural abnormalities also, like deletion or duplication. Now you can see in this segment, this is one chromosome, a part of this segment is deleted. So this is deletion. So this can also occur. There could be some insertions, means you can see in this one segment from this chromosome has inserted into the other chromosome. So this is insertion. There could be inversion also. So what does inversion means? Ideally, it is like this, but you can see this star has gone here and this has gone here. The whole segment has inverted and position of centromere has also changed. So this is an example of inversion. So these I am discussing about the structural changes which can also occur in the chromosome. And last is the ring chromosome. In the ring chromosome, the end of the chromosomes, they are broken down and they join to form a ring. So this is a ring chromosome. The other important structural malformation is this translocation. Translocation means a part of segment of one chromosome has gone to the other and the other one has gone here. So there is mutual translocation of segment. So it can be balanced, it can be unbalanced. Balanced, you can see in this, there is no gain or loss of material. Only this blue part has come to the purple, purple has gone to the blue. So this is a balanced translocation. But there is a specific type of translocation, which is Robertsonian translocation, which basically involves your acrocentric chromosomes. So what are these acrocentric chromosomes? They are 13, 14, 15, 21, 22, where we have a long arm and a very short P arm. So you can see in this, it has only 121 chromosome and this 21 has shifted to 14. So this is 14-21 translocation. So this was in the father, and father will be absolutely fine because 2 chromosome 21 are there, only that the 21 chromosome has gone to the and stick to the 14. So this is a balanced type of translocation. But when gametes are formed, it is likely a possibility that the fetus or the conceptus can have unbalanced genomic imbalances, which either deletion or duplication. So you can see in this picture, there are 221 and 121 is attached to 14. So this is trisomy 21, which was given arise due to the 41-21 translocation. So whenever you have a couple with recurrent abortions or any previous history of any bad obstetric history, chromosomes must be ruled out to see whether there is any translocation in the couple or not. So about 50% of the miscarriages have chromosomal abnormalities. At birth, it is very important that risk of chromosomal abnormality can be up to 1%. That means out of 100 births which are occurring, one of the baby can have chromosomal abnormality. So it is very important to discuss all these aspects with the couple. Now variants can affect DNA also. Now we have discussed about the whole genome, we have discussed about the chromosomes. Let's come up to the DNA. DNA, I told you, basically it consists of genes which have the arrangement of bases. So it is the bases which are affected in the DNA. Okay, so this is a very simple diagram. So this is the DNA. This is GCA is the sequence of bases, and this is one amino acid which is coded by these bases. So this is a normal sequence. Okay, synonymous means silent. Okay, silent. In this, instead of G, T has come, the protein remains the same. It is known as the silent or synonymous mutation. But generally, it doesn't harm so much. Then there are non-synonymous mutation. Non-synonymous means that there is some other protein is formed. It can be missense, where you can see instead of G, the C is there. The amino acid has changed. Okay. So it alters the amino acid and protein sequence. There could be nonsense change. Nonsense means just rubbish. Rubbish in this sense means if G is replaced by T, it is TAA, stop codon, and protein function stops. Okay, so this is a nonsense or stop codon. So in gene, we generally look for these types of point mutations, which are occurring at the level of bases. Then there could be some splice site changes, promoter changes, or enhancer changes. Now let's come to apart from point mutations, there could be some deletions or insertions of base pair also. Okay, suppose you can see in this case, this T is deleted and this CGC, the whole sequence has changed. Okay, so they are known as the frame shift mutations. So you can see even one deletion of a base has led to such an effect that the whole sequence has changed. Okay, but sometimes if it is in a multiple of 3, that means if multiple of 3 are there, then the protein may be shortened, but sequence can remain the same. So these are the changes which can affect the DNA. Another very important thing for you to do, you must be seeing all the reports in your daily practice where there is written heterozygous, homozygous. What is all this? First of all, wild type. Wild type means natural form, the alleles when they are present in the natural non-mutated form. Okay, so let's see this. So the 2 alleles are present in the natural form or normal form. This is wild type. Heterozygous means when the different alleles are present on both the chromosomes. Let's look at this. So these are the 2 genes, alleles. One is normal, one is abnormal. We call it as heterozygous. Homozygous, when both the alleles, they are same, they can be normal or they can be abnormal. This is homozygous. Now, what is this compound heterozygous? Compound heterozygous means the abnormals are there, but one abnormality in one gene and another abnormality in the other gene. This is compound heterozygous. And hemizygous, we use the term for any X-linked disease in case of males. So this is the report which I am just sharing with you. Like in this case, you can see it is the homozygous variant. Okay, homozygous means this variant, abnormal variant, is present in both the alleles of a gene. Now you can see this is one gene. It has one variant, abnormal variant, and other abnormal variant. Okay, so that's why they have used the term compound heterozygous. Okay, now in this case, you can see it was autosomal recessive disorder. This is husband, this is wife. You can see they are present in the heterozygous forms, means they are the carriers. So don't get confused. Okay, so in case of autosomal recessive, it can be homozygous or it can be compound heterozygous also. Now, apart from this whole genome, chromosome, DNA, there are other terms also which includes epigenetics. Okay, so epigenetics is something different where the gene expression, changes in the gene expression, they do not occur due to the change in the DNA sequences. Rather, there are certain other factors which control the gene expression. Suppose you study that gene, that will be absolutely normal, but there are certain promoter sequences, regulatory elements whose defect that is controlling the effect on the gene. So that is epigenetics. Then there could be some triplet repeat nucleotide disorders also, like Fragile X, myotonic dystrophy. Whatever you do, whole exome, chromosome, karyotype, you are not going to find anything because they are the totally different set of disorders. Another term, mosaicism. Mosaicism generally means when we have 2 cell lines, one is normal cell line, but few cells, they undergo mutation and then they expand. So abnormal cells are also there in the organism. So we use the term mosaicism. Mosaicism can be somatic when it includes the abnormal cells only in certain organs, or it can be germline. I'll discuss this germline mutation ahead. Quickly, let's come to the Mendelian modes of inheritance, which is mostly for the monogenic disorders: autosomal dominant, recessive, X-linked dominant, X-linked recessive. Let's come to autosomal recessive, very common when the phenotype is expressed only in the homozygous form. So this is the normal. When we have 2 abnormal alleles on both the chromosomes, they will have the disease. Most common, our hemoglobinopathies, cystic fibrosis, all our inborn error of metabolisms. Very important in this case, males and females are equally affected. If you see a pedigree, this is a pedigree where they suppose father, mother, and they have 4 children. So you can see their 2 children are affected and they are carriers. So those affected individuals, they are in the one generation and they are siblings. Okay. So if a couple comes to you where both husband and wife are carriers, then you can see this is one father, this is mother. We know 50% comes from father, 50% comes from mother. So there could be these permutation combinations. We all know this. And there are 25% chances of recurrence, 75% chances that baby may be normal or affected, a carrier. Another is autosomal dominant, when the phenotype is expressed in the heterozygous form. Means even if one allele is there, they will affect the individual. Most common tuberous sclerosis, ADPKD, neurofibromatosis, Marfan syndrome. Again, both males and females are equally affected. They are not X-linked. But in this case, you can see this pedigree. So in this pedigree, in these multiple generations, you can have multiple affected members. Okay. There is a specific term which is used for these autosomal dominant disorders is penetrance and expressivity. Just try to understand this concept. Penetrance means, suppose in 100 in a population, it may be possible one is normal. I told you it has autosomal dominant mode, means only one need to be abnormal. But it is not necessarily out of those 100 individuals, everybody will have the disease. It may be possible only 90% have the disease, 10% do not manifest the disease. So that is actually the penetrance. So penetrance with the predisposing genotype who are actually affected. So it is an all or none phenomenon. If it is less than 100%, we call it as incomplete penetrance. Okay. So for example, you can see in a family, father may be completely asymptomatic and child can be so severely affected with neurofibromatosis. So this is incomplete penetrance. Expressivity means those who are expressing the disease, they will have different grades of expression. For example, father can have
Ma'am, sorry to interrupt you. Your time is up, ma'am. 20 minutes
I'll
off.
just take 2 minutes.
Yeah.
And this is expressivity. So risk of transmission is 50%, as we have seen. So it will be affected parent, one has mutation, and there will be 50% chances that it may be transmitted. So one case I'll just briefly discuss. So a couple comes to you with previous baby affected with tuberous sclerosis and mutation is confirmed. Okay. So you see the parents, case scenario one is both parents are completely asymptomatic with no stigma, but father has the mutation. So that is incomplete penetrance. Then second scenario is father has very mild adenoma sebaceum and has the mutation, that is expressivity. But scenario 3 is very important. Both parents are completely asymptomatic, no stigma, you did the testing and both of the parents were not even carriers for the mutation. But you assured them that nothing will happen, but second baby is also affected. That is the concept of germline mosaicism. Germline mosaicism means it may be possible that the mutation is present only in the ova and the sperm. So recurrence risk is never 0%, it is 1 to 2%. Secondly, now quickly I'll just take 2 more minutes. X-linked recessive, when they are expressed in all the male heterozygotes because they have only one X chromosome, the correct prototype is hemophilia. Okay. So in this case, you can see all the males are affected. And if suppose the husband comes, nowadays with the treatment, the hemophilias are surviving, they are marrying. If he comes to you and wants to ask, will my children be affected? You have to just say, boys will be not affected because he will give X chromosome to the daughters. So daughters will be the carriers. However, if mother is a carrier, then she is likely to affect 50% of the sons and daughter will be a carrier. Last is X-linked dominant. X-linked dominant may both males and females will be affected. Female will be severely affected. It is very rare, generally not seen, vitamin D resistant hypophospholipids. In this case, male will transmit to all the daughters and female will transmit to the daughter and the son. Y chromosome, very, very few genes, only SRY gene, which is the test determining factor, and then can affect the infertility. Otherwise, male baldness is typically associated with Y chromosome. So learning points, genetic variations can affect either the chromosomes or the DNA. Exceptions are there. Mostly monogenic disorders, they follow the Mendelian mode of inheritance, which is important to predict the risk of affection, risk of recurrence in the next pregnancy. But still, clinical diagnosis is always supreme. Thank you so much. I had to
Thank you very much,
go.
Dr. Sakshi. I think that was a in the short period of time, you really covered a large, as very important translatable parts of the topic. Thank you so very much. So
Thank
I
you.
think there is a paucity of time. We will keep the questions for the end of the session if possible. So over to you, Dr. Meena, to introduce the next speaker, please.
We are not encouraging questions because that way I give out the answers for the post-test.
Okay, so that's all
Yeah,
good.
that will all be kept for the end.
Great.
Thank you.
Sure. So Dr. Meena, please.
Yeah, good evening, everyone. After that very lucid presentation, we have yet another important aspect of medical genetics, understanding. The Dr. Sakshi just touched upon it, but we're going to learn more about the pedigree from Dr. Shagun Agarwal, who's professor, Department of Medical Genetics, NIMS, and she is DM in medical genetics, numerous publications, 78 and 11 chapters and books. She's also associate editor of the genetic update for the next generation clinicians, and she has so many professional achievements. An area of interest are the fetal dysmorphology and reproductive genetics. So over to you, Dr. Shagun.
Thank you, Dr. Meena. So I'll just share my slides.
Dr. Shagun, before you start, I was just curious to know, are you a PG from UCMS?
Yes, ma'am. Actually, I didn't want to
Okay.
interrupt, so I thought I'll send you a message privately
Okay,
after my
great to
talk.
see you. No, I
It's so
wasn't
nice
too
to
sure.
see you. It's so nice to see you
great.
after so many
Ditto.
years.
Same here. Good to see you.
So my slides are visible?
Yes.
They are. Yeah.
Yes, they are.
My dear. So after that wonderful lecture by Dr. Sakshi, who's given you some basic overview of genetics. So my talk is going to be more clinical. So I'm trying to cover in this talk how for an obstetrician it's very important to take a family history. And not only just take a family history, but learn how to draw a pedigree. Because pedigree is something which is available to literally everybody. It's a bedside tool and it just needs a pen and a paper, and it can really solve so many mysteries about your patient, about diagnostic dilemmas at times, and also it saves paper, so it's a go green way to practice medicine, I would say. So let's see the scenarios in a bit. So this is like, sometimes as obstetrician, and I also know when I mean primarily an obstetrician, we don't think so much about genetics, and there are certain scenarios where we don't even think of doing any genetic test. But then, of course, now myself, having gone through a genetics residency and practicing now for many years, I realized that practically every scenario or every situation that a OBG practitioner comes across has a genetic component to it, and we just need to stay open, keep our eyes open, our antennas always, in the receiving mode to be able to catch that patient needs further workup. So these are, I mean, very common scenarios which everybody is seeing day in and day out, where there would be a relevance of genetics, and for that to be able to find which patient needs that special attention or a genetic workup, we need to basically do the pedigree drawing. That's the simplest way that by which you can detect a patient who needs a genetic evaluation. So here some of the scenarios are like more relevant, of course, to obstetric practice, and some are more for, I mean, the gynec component of it. So this is just to represent what all scenarios we should be thinking of a genetic cause. And then I'll straight away go on to the main topic, which is drawing of pedigree. So basically, a pedigree is helping us to represent the family history. That's what it is. So it's a kind of a diagrammatic representation, so that
what would probably require you 4 pages to fill, and which is something which is not possible in practice. And then going back and reading those 3-4 pages again is not possible. So a pedigree really solves all these issues, whereby you just need to learn some basic symbols and start using them, and just by another probably some 20 patients, 30 patients down the lane, you will be able to do it like a pro. So it's very simple, and it's a very easy way of representing the family, and gives you so many clues. So the word typically actually as such comes from an old French phrase, and this literally means the foot of a crane. I mean, kind of just representing that, how the foot is structured and has these, the various toes or whatever claws, whatever you call them. And then if you go back to some older manuscripts and all, we can sometimes see that something similar to what we now draw as a pedigree is actually being used to represent ancestry. So, so how does it help us? So this is just I've just listed point wise, but I will take you through some case scenarios to emphasize these points. So, as I mentioned, so this pedigree is basically a graphic depiction of how various family members are related to one another across different generations, and even of course in the same generation. And also besides their relation, it also reflects, or it is able to depict their disease status. So by drawing a pedigree, we can get a lot of things. So once you have it in front of you, you can make out that probably your patient has a genetic condition. So it can flag a genetic disorder. Then it can help you in recognizing what could be the potential inheritance pattern. So just like Dr. Sakshi showed, so the various inheritance patterns, autosomal dominant, autosomal recessive, X-linked, recessive dominant, all those become clear once you sit and draw the pedigree. And then once you have made that, then you can also find out who are the family members who are carriers in the family of a particular genetic mutation, and who have a increased reproductive risk of having a child with a genetic disorder. And then these individuals can be counseled, and you can intervene, prevent a genetic disorder from happening in their offspring. And of course, we can also, same pedigree, we can use to screen other people in the family, and also decide who needs to undergo the genetic test, who doesn't need to undergo a genetic test. So there are so many information which can actually come through that. So there are certain basic symbols, and these are very simple. There's nothing like very confusing or, very complicated about this. So we use these squares and circles. Squares are basically males. Circles represent female. And then typically an unaffected person, the square or the circle remains empty. So it will be transparent basically. So this is a normal male, this is a normal female. And when somebody is affected with a disease, then we shade it. So like here, it's shaded. So affected male, affected female. But then if you see here, we are having, one person, one square having 2 shadings. This one circle has 4 shadings. So actually here, basically, this is sometimes an individual can have more than one disorder, or sometimes it is the same disease, but the manifestations are varied. And unless you make a diagnosis, you think that these are different diseases. So in that case, we shade the different symptoms or the different presentations, or sometimes different diseases. If they are really 2 or 3 distinct diseases, so the different shading is given to a different condition basically. So if there are 4 conditions, then basically you, like this lady, she has 4 different conditions. This individual has 2 different conditions. And then typically at the bottom, we'll make a legend where we will show that this shading means so and so. So then besides this, then of course you will see other things. So sometimes you have you can put a number within the circle. So when you want to show that how many women are there, like suppose you are drawing somebody's sister. So how many sisters are there? We can put some numbers inside. Sometimes you don't know the number, so we can write an N. Sometimes the gender is not known. Then we draw a rhomboid like this. So these these basic some symbols can be there, and of course then this is the line, the diagonal line, which represents a deceased individual. And then below that we usually write D. D is for death. Dot at what age it happened. So 35. So these are some of the starting points, and here we can see this line joining a circle and a square. So this is a marriage line or a relationship line in today's era. It not always be marriage now. So yeah. So then after these basics, then we need to remember two more important things. That is the concept of a consultant and the proband. So a proband is typically a person who is affected. So a proband will always be shaded. And the proband is sometimes in one family, there can be more than one person having the same disease. But in that case, we usually, proband is indicated as a person who first was diagnosed, who came to the attention first. So we usually put one proband in a family, but sometimes you can have a family coming with two children simultaneously. So then you can indicate even two probands. So proband is indicated one, of course, by shading. And then you put an arrow and then you put the letter P. So this is the proband. This is the affected individual who first came to medical attention. And then now this individual here you can see has a son who is also affected, but he is not the proband. He was secondarily diagnosed. And then you have the consultant. So consultant is a healthy person, but they have consulted you because they are concerned that somebody in the family has a genetic disease. So they have come for a consultation and they want some evaluation for themselves or for that other person. And then they want subsequent counseling and what next to be done. So here, like you have this couple. So, I mean, this individual is the one who came for an OP consultation, and they have most likely come because they have two children having some disease. And then they also have a healthy child. So they may be looking for maybe reproductive options or some other options. So this individual has come. So this is a consultant. So, and the consultant is indicated by a C. So an arrow and a C. Then we can have certain other symbols, especially for reproductive scenarios. So like one, this I mentioned, undefined gender. So rhomboid with nothing inside the rhomboid. Then we have, if we write a P inside a rhomboid, this is a pregnancy with an unknown gender again. So in our country, it will always be this. This is, of course, I mean, from the classical, I mean, the Western literature, you could say. So there, if the gender is disclosed for a pregnancy, then you can draw a circle with a P or a square with a P. But for us, of course, it is always this. A rhomboid with a P inside is a pregnancy. And then you have another, a diamond. Rhomboid or a diamond. And then you have this another symbol, which is a triangle. So now a triangle is basically, again, a unshaded triangle and a shaded triangle. So an unshaded triangle with no line across it is a spontaneous miscarriage. Whereas if you have a line across it, it is a MTB. Therefore, and whatever indication, but the fetus as such is not really affected with it because you're not shading it. Whereas if you see here, this is a 17 weeks. So this is a shaded triangle. So this is a fetus which got aborted and which is affected with cystic hygroma. So it is shaded. And this is a fetus which was terminated because, so the line indicates termination here. Terminated because it was affected with trisomy 18. So again, affected status is by the black shading and the line indicates that it was a medical termination. And then ectopic can be shown like this. And then stillbirth, if the gender is known, then by a square or a circle. Gender unknown means again we come back to the rhomboid. And then we draw a line across to say that person is deceased and to distinguish it from a pediatric or an adult patient who's deceased, we write SB. SB is stillbirth. And then number of weeks below, showing that at what weeks the stillbirth occurred. And then this is, this is deceased, not diseased. So deceased individual where you can show the age of the death. Is the death at 35 years, death at 4 months age, death at in the 60s. Gender not known here. This is a female, this is a male. Because they are not shaded, so we are assuming they are not affected with any specific genetic condition. So these are the individual symbols, and then you have these lines. So the line, one line is the horizontal line. So horizontal line is a relationship line. So a single line is a non-consanguineous couple, a double line is a consanguineous couple. And then if you have an interruption like this means that the relationship no longer exists. And then you have a line of descent, which is a vertical line. So from a horizontal line, if there is a vertical line moving downwards, this shows basically the offspring of this couple. So this is what is shown here. And now again, this lady is married. So this is a married line, married to this individual. And then now these are their children. So again, a vertical line means their descendants. And in their descendants, you have now again further, they have 3 children. So then it is made in this way. The 3 children are represented. 2 boys who are healthy and 1 girl who has a disease. So she's shaded and she is the probe and she was the one who was brought for evaluation. And then this line, basically again the vertical line. So it's not to be confused with the offspring line. Offspring line runs directly down from the marriage line. Whereas the sibling line basically, so this runs down from, so this marriage line, after that it divides, if there are more children, into another horizontal line. And from that, whatever is coming is basically the various children of that particular couple. And this is basically the sibling line now. So then you have this relationship line, the line of descent, the ship line, this horizontal, and then the individual's line. And then there are some more symbols which are important for OBG practitioners. So to show twins, again, monozygotic twins, which are joined here, dizygotic, zygosity not, trizygotic twins, parentage not. So you put a question mark here. Then a couple who has chosen voluntarily to not have kids, represented by the descendant line ending here with a small horizontal line. It could be a voluntary vasectomy or a tubal ligation, which can be written below that line. Whereas if you have infertility, then you put 2 lines over here. And then if you know the reason, then you write the reason below it. So fertility because of azoospermia, infertility because of endometriosis. And then there are certain symbols for even adoption. So adoption in the family, adoption out of the family, adoption by a relative. So there are various symbols. There are now some symbols also for the various ART scenarios which can be used. So for the purpose of genetic evaluations, nowadays very often patients do have genetic reports also available. So that also can be included in the pedigree instead of writing lengthy notes about that. So for that, we can basically, here like you can see, so basically this is a person. So if this person has been evaluated, then you put an asterisk. And what was the evaluation is indicated by an E like here. So E and whatever was done is indicated next. So, and whether it's positive or negative is indicated here. So like here we are having a patient who is basically a carrier as reported by the patient here, it seems. The asterisk is not there because not evaluated by us or ourselves. Somebody has told that the patient is a carrier. We need to still check the report. If we check the report, we can put the asterisk here. And then here we have a lady, 25-year-old lady, who has a mutation report. So she is already positive for a BRCA1 mutation. So that is written here. So this E is for evaluation or evaluation. So evaluation positive for this BRCA1 mutation, and evaluation 1 is a mammogram, negative mammogram. So right now she doesn't have any lesion. And her mother was, it seems, affected with the breast cancer. And this vertical line is indicating basically a person who has a mutation but not affected right now. So same way here we have some individual examination negative, mutation showing some report which is mentioned here. And similarly, so we can include lot of things in a pedigree so that in a very concise manner, we can actually represent everything practically in the sense that we can represent the whole family tree up to 3 generations. We can put every individual in the pedigree. We can show who's affected, who's not affected, what disease they are affected with. Is there a genetic report that can be included? So all this can be there in a single pedigree, which takes hardly half a page. So usually, I mean, just to remember to like want to start drawing a pedigree. So mostly we start with the proband and then we start drawing the relatives of the proband. And for consanguinity, we need to remember how to connect the people who are related to each other. So just this is just a very simple pedigree of which is complete pedigree. So complete as in all the components have been taken care of. So we have an individual who is affected with cystic fibrosis. So we have the lady is shaded and then the legend is written here. So what does red color mean? It means cystic fibrosis. And then this arrow is there, which means she's the proband. Then we had showing the various generations of family here. So this is her husband. Then this is the, I mean, her husband's parent, her parents, and their child. So these 1, 2, 3 here in Roman numerals indicates the generations. These 1, 2, 3, 4 here indicates the individuals in each generation. So like that, I'll not go into this too much. So by looking at pedigree, because Sakshi has already covered this part, we can also immediately make out the inheritance pattern. So here we are seeing a disease which is running across generations. So vertical pattern, this is autosomal dominant disease. Here we see a disease where the partners are carriers and their children are affected. This is an autosomal recessive disorder. Here we see a pedigree where only males are affected and females are all having this dot in center, which indicate they are carriers. So males affected, females, unaffected females being carrier. This is X-linked recessive, and rarely some conditions can have X-linked dominant inheritance. So what we need to remember is that an X-linked disease will never go from an affected male to his male child. That is the operating principle here, because a male cannot transmit his X to his boy. He will always transmit his Y chromosome. So X will always come from the wife. So X-linked disease will never get transmitted from a male to his son. So now quickly going through some very simple cases, very common things, but just trying to see how just drawing a pedigree solves your case and you know what to do next. So here we are seeing basically, so very often you will have a couple and they'll come and they have some family history of a genetic disease and now they are worried that their child could also be similarly affected. So here is one such couple. So very often we will just ask, okay, who's affected? What is your relation to that person? And then you will write, okay, some cousin is affected, or here, like we are seeing here, the nephew is affected,
I'm
husband's
sorry to interrupt you.
nephew
Your
is affected.
time is up.
Yes. Did I not start late? Started with, sure. Okay, fine. I'll just take 5 minutes then. I just have some pedigrees to show. That's all.
Okay.
So here we can see, like by drawing the pedigree, I can make out that this nephew is basically the husband's nephew. So, and then the disease he's having is Duchenne muscular dystrophy, which is an X-linked disorder. So this child basically could have got the disease due to a new mutation or because of lady being a carrier. But this person cannot be a carrier because he's a male. If he had the mutation, he would be affected. So which means now that this couple are not at risk of having a child with Duchenne muscular dystrophy more than the background risk. So there would be a background risk always. So if I was just going by the premise of there is some family history, a nephew is affected, I would be really concerned. But once I draw the pedigree, I can see their relations and I can see that this pregnancy couple's child is not at risk. So now a similar same scenario. So here there is again history of a neuromuscular disorder. History is very much looking like Duchenne muscular dystrophy, which is an X-linked recessive disorder. So we can see only males are affected. They are connected through all these female. And again, this couple has come. So now they want to know their risk. So the first thing we have to see whether it is on the mother's side or the father's side, because we are now looking at an X-linked disease. So here we can see it's on the wife's side. And then we can connect who all are related, how. So by connecting all these individuals through a pedigree, we find who are the obligate carriers, like the women who are surely having the mutation. So we find she's an obligate carrier, she's an obligate carrier. This lady is at 50% risk of being a carrier and our patient is at 25% risk of being a carrier. So by drawing a pedigree, you find this and then you do the genetic test for the obligate carrier because the child is not alive. And here that's what was done. We did the test for the obligate carrier. We found the deletion And then now after that, we were able to test this lady and we found that she is also a carrier. Now this lady has a risk of her male offspring being affected with DMD and she needs a prenatal test. So just drawing of pedigree helps you doing all this thing on finding whether there is a risk, quantifying the risk, finding whom to do the genetic test for, and then counsel accordingly. So again, it is a very, very short case. It's just 2 minutes. So this again, this couple has come because there is a family history of Down syndrome. But once we draw the pedigree, we can see that it's a husband, sister's child who has Down syndrome. There is a report which shows it's a free trisomy 21. So this would have arisen because of non-disjunction in maternal meiosis. So this has got absolutely nothing to do with this couple's risk of having Down syndrome. So we can counsel them that they just have a background risk. Whereas if you see this couple, they came because again for the same thing, their first child, and this was aborted, was found to have some scan abnormality. Later on, amniocentesis is done. Child found to have Down syndrome, diagnosed by doing a FISH. Now they are 6 weeks pregnant and they want to know their risk. So here now when we look back, so FISH is just going to count the 21 chromosomes, but when we do a karyotype for the child, we find that the child actually has 1421 translocation and also has 2 normal chromosome 21. So then we do the karyotype of the parent and we find that yes, the husband also has a 1421 translocation and 1 normal chromosome 21. So now the recurrence risk for this couple jumps up to as high as up to 15%. So this couple really needs counseling and they do need invasive prenatal testing. There is no role of doing an NIPT here because the risk is too high over here. And the other important thing is now husband siblings also need to undergo karyotype. You can see how his brother, his wife has had 3 miscarriages. So probably he also has the same translocation. So finally, a non-ob scenario of a lady who comes with secondary amenorrhea, hypergonadotrophic hypogonadism. Just taking a simple family history, we find she has 2 brothers with intellectual disability. And for a geneticist, for me, this is an immediate indication of realizing that this could be Fragile X3 mutation, which is there in this lady, and brother have Fragile X syndrome, and that's what it turned out to be. Both these brothers had Fragile X syndrome and she had a Fragile X3 mutation. So in fact, 15% of VOF patients who have a family history of VOF or intellectual disability in male members have this pre-mutation, and even 2 to 5% sporadic patients with VOF have this pre-mutation. It is important to pick it up because tomorrow this lady may reproduce because many of them have secondary amenorrhea, and then her offspring could again have Fragile X syndrome. So the key takeaways from this would be that one thing again, just to say that I think it's important for everybody to learn pedigree drawing. It's a really simple thing. Once you practice, after 20, 30 patients, you'll be able to do it. It's very, it just needs a pen and paper. It's a simple bedside tool and it will give you a lot of information which will be very helpful. And it's a very kind of a clear, concise, and also comprehensive representation of a family tree, and it very often helps in finding that patient who needs a genetic workup, and thereby by doing the subsequent workup, it enables your counseling, it enables you for further management.
Thank you, Dr. Agarwal. Thank you, Dr. Agarwal. Very nicely, the plotting has been, explained by you, and I think everybody is going to be benefited. And what we really need is to bring it into our practice. And I know some people who are already into practice, of course, all the genetists do it anyway, but in routine obs and gynae, especially the high risk, they should make it a point. And yeah, there are the younger ones I find who are joining, they it's in their pen. They do it, but the much senior ones, they have to make an effort to do it. But this is something really so nice, and everybody, I think they can do it, and we have to do it. Thank you very much.
Thank you.
So let us move on to the next session of today. I would like to invite the first chairperson for this session, Dr. Seeta ma'am, who is one of the conveners of this certificate course. She is a consultant fetal medicine and obstetrician and Chairperson of Genetics and Fetal Medicine Committee of FOGSI. Another chairperson, Dr. Shobha Goody ma'am. She is professor and HOD at St. Philomena's Hospital, Bangalore. And ma'am also holding prestigious positions in FOGSI as well as ICOG, and also has been honored with many awards and several publications to her credit. And her special interest is in, I can't see the below the slides.
It is
but,
okay. It is okay.
Okay.
Thank you so
Hello,
much.
ma'am.
Yeah.
Over to you and Sita, ma'am.
Yeah. Sure.
I'll
Yeah,
introduce-
Sita, you can introduce the
Yeah.
first speaker. Yeah.
Thank you. Firstly, thank you to Dr. Sakshi and Dr. Shagun for those brilliant startup for the overview of genetics and pedigree charting, and to the chairpersons, Dr. Radhika and Dr. Meena. And it is now my proud privilege to introduce to you another doyen of clinical genetics, Dr. Chaitanya Datar. He's a consultant clinical genetist at the Director Sujanan Clinic and Bharatiya Vidyapeeth Medical College Hospital in Pune. A doyen in genetics as well. And today we are going to be talking about chromosomes and single gene testing. A lot of the obstetricians, general obstetricians get confused as to when exactly to ask for which test and which test represents what. I think these two lectures will actually clear all the doubts. And so over to you, Dr. Datar, for your talk on chromosomal testing. Thank you.
Thank you, Dr. Sita. At the outset, let me thank the organizers for this opportunity. And so let me begin right away. We are talking about chromosomal testing today. So essentially, what was in the way back in 1956, it was the first time when the
Sorry to interrupt. Can you just make it full screen, Dr. Datar?
Oh, I'm sorry. Yes. So in 1956, it was first known that all humans have 46 chromosomes, and since then we have come a long way. And the initial tests actually, wherein the initial, I would say, headways were mainly in the cytogenetic or chromosomal domain. So wherein the karyotype came in, then FISH. Now we have arrays and non-invasive testings. So always the question is that do these newer tests really replace the older ones, or are they complementary? And obstetrics is one arena wherein cytogenetic testing holds a major role, and in almost all sorts of indications like infertility, pregnancy losses, product of conception testing, when we have abnormal antenatal scans or marker reports or anomalies, all these places cytogenetics plays a very major role. So we need to really understand the role of cytogenetics in the obstetric domain. Now, this, if you look at these 46 chromosomes, they do constitute our genetic. This is the lowest resolution of the whole genome, actually, if you say. But if you open up the chromosome, it is going to be a long thread of DNA. So if the DNA, if you coil and supercoil, it forms that compact structure called chromosome. So disorders can be at different levels. So if the abnormality is at the chromosome level, we are doing a karyotype and reviewing. So if you open up the chromosome and look at the segments, that is when you're doing a contiguous gene syndrome evaluation, either by a chromosomal microarray or by FISH. If you're going to the rung of the ladder, that is a single gene testing, which Dr. Ashwin will be discussing later, we are talking about monogenic syndromes. So essentially, it's like the scaling of the genome. You're going from a lowest resolution to the most deepest part. So chromosomal evaluation, then you're looking at copy number variations, that is segments of DNA which have gotten deleted or duplicated. And then further down, it's going to be the ATGC bases that you're looking at. So essentially, today we are going to, my talk is going to be restricted on chromosomal analysis and CNV analysis by microarray. So if you're looking at the most basic test, it's the karyotype. Karyotype is usually evaluating for common aneuploidies in fetuses like Down syndrome, Turner syndrome. And very importantly, it looks at balanced structural rearrangements, which is not looked at by any other test, that is translocation, inversions, etc. So if you look at the karyotype, these are called G-banded karyotype, GIMSA banding, and you're seeing these light and dark bands. The light bands are the euchromatin, dark bands are the heterochromatin, and the chromosome has its P arm and a Q arm as well. And various segments of the chromosomes are appropriately labeled. So especially when you're looking at cases when there are translocation, they'll give you a chromosome number, so which will indicate the point at which the rearrangement has happened. So essentially, even the banding techniques are important. And when you're looking at the banding technique, you can see the same chromosome, but see the resolution. So if you're looking at the same chromosome, if you're looking at it in a pro-metaphase or prophase, it's a more longer chromosome. So a higher resolution banding is usually desired in case we want to look at, structural rearrangements in the chromosome. Small, stubby chromosomes with poor banding pattern will usually not give you a proper and correct diagnosis. So banding techniques are very, very crucial. And this is something called ideogram. So we have these chromosomes wherein the banding pattern is very typical. And then based on these ideograms, they are classified into various numbers and various groups. So the 22 pairs and the sex chromosomes. Now, banding techniques have evolved, and rarely we do use C banding and North staining, especially when there are prominent satellite regions in some of the chromosomes. There sometimes you may have to use a C banding or a North staining in some of these cases, but it's not routinely used. The routinely used technique is the G banding technique or the GIMSA banding. The second test in cytogenetics that is utilized is a FISH test. It's called fluorescence in situ hybridization, and that's commonly used for rapid aneuploidy screening in pregnancy. That is, you want a quick result. The screening test has shown a high risk for Down syndrome, and you want to rule it out Down syndrome quickly. At that point in time, you can utilize a FISH, and it can also be utilized for certain microdeletion cases, like you're suspecting DiGeorge syndrome or an Prader-Willi-Angelman. In those cases, we do utilize FISH. But for FISH, remember, we need to know the specific locus because it's only going to amplify that specific region. So the probe is going to be, the DNA in that segment is denatured. A fluorescent probe is used, and under the fluorescent microscope, only that specific point will be highlighted. So you can identify if that particular region has gotten deleted or duplicated. But now assume that we want to look at the entire genome, the deletions and duplications in that, for which we will require a chromosomal microarray or a cytogenetic microarray. Now, this is a technique which is usually used for evaluation of fetuses with anomalies, soft markers. You can also utilize for product of conception studies for pregnancy losses. So if you look at the slide below, you can see that a small segment of the chromosome has multiple regions which are tiled like this. And based on the hybridization of the patient sample and the actual probe, you will find the areas which are deleted or duplicated. So here what you can see that there's an area of deletion which is noted in this particular segment. So this is going to look at a step deeper than the chromosome. So overall, if you're looking at a karyotype, the resolution is 5 megabases, while if it's a microarray or a molecule, it can be as high as about 150 to 300 kilobases. On an average, it gives about 15-20% more information than a baseline karyotype. So if you enlist the genetic tests that are used in obstetrics, the first three, that is FISH, karyotyping, and chromosomal microarray are cytogenetic testing, which we are focusing today, while PCR-based techniques, Sanger, exome genome analysis are molecular techniques, which will be discussed later. Now we'll go to some case scenarios from obstetrics. So I thought that we will review it from an obstetric perspective. So in case a couple has come with an infertility or recurrent implantation failures, so which is a test of choice in these patients, it is going to be karyotype. So what do you expect in these couples? Either there could be an extra or missing chromosome, like a rare case of aneuploidies, or there could be structural rearrangements, translocation. Remember, microarray is generally not useful and should be sparingly used in cases with recurrent implantation failures or infertility. So what do you identify in some of these? So you know that 46 chromosomal complement is a normal complement. So each a pair of chromosomes from 1 to 22 and sex chromosome. Now sometimes you may find males with infertility having Klinefelter syndrome, an extra chromosome, or females with variable Turner syndrome also you can identify. But in some of the cases you see here, like there are two X chromosomes, but if you look at it carefully, both the chromosome in this chromosome is a little longer, and both the arms of this are Q arms. Actually, this is called isochromosome XQ, which can give rise to a Turner-like phenotype and sometimes present as an infertility. So you utilize this metaphase FISH to identify that this is an abnormal X carrying both the XQ arms, and there's no XP arm here. Or sometimes there could be a ring X chromosome which you identify in a karyotype, which can give rise to an infertility or a pregnancy loss like situation. Now, in males also, you may get something odd like this. For example, this is a karyotype, but you're finding extra chromosome. This is a marker chromosome. Now, there is an history of infertility with 2 IVF failures, and this karyotype showed a marker chromosome, which needs to be defined further. And when we use this colorful technique called spectral karyotyping, we identify that this particular region or this extra chromosome belonged to chromosome 15. So this was a derivative 15. So the husband had this particular thing, and therefore it was causing the recurrent miscarriages. So obviously we could give them the option that they can consider a PGT or consider a donor semen for this particular case. So obviously when you identify the etiology, you are able to counsel the patient as regards to their further mode of conception as well. Now, sometimes you may face in this particular case, this is an individual who underwent a karyotype. If you look at it carefully, it is a 31-year-old male patient, and you see the karyotype is 46XX, right? So this is an XX male, but if we did a FISH later on the same sample, and what we found that this male on one of the X chromosome has an SRY gene which has translocated. So essentially, this is, if you're looking at it at a genomic or a genetic level, this is an XXY pattern, and therefore this individual has normal male genitalia but shows spermatogenetic arrest and severe testicular atrophy with azoospermia. So evaluation of a karyotype in cases of infertility as well as recurrent implantation failures is very, very crucial. Right. Now, see this case. There's a period of infertility of 6 years with poor semen quality. This couple attempted 6 IUIs, 3 IVFs with poor blastocyst formation. And at the end, it was found that the male was a carrier of a translocation between chromosome 1 and 2. So if a couple karyotype would have been done in the initial stage, we could have given them the option, of a PGT or possibly a donor gamete, and, would have prevented so much of costs as well as, so much of mental trauma or agony to the patient. So doing a couple karyotype at the initial stage of infertility cases or recurrent implantation failures is very crucial. The second problem that we face in obstetrics is recurrent pregnancy losses. Again, here the karyotype is a test of choice, and what we expect here is a balanced chromosomal rearrangement. Again, microarray should not be considered in couples in these cases. Now see this case, there's a case of primary infertility. They underwent 2 IUI pregnancies but ended up in a miscarriage. Husband had a low semen count with poor motility and high DFI. They were planning an IVF ICSI conception, but a karyotype revealed that there's a translocation between chromosome 3 and 22. So obviously now the outcomes are going to change for this particular family, and therefore always remember semen does reflect chromosomes. So do a karyotype in all males wherein there are semen quality or quantity related issues. Then you sometimes get karyotypes like this is called Robertsonian translocation, when instead of in this particular family, okay, there's a translocation. This happens usually with acrocentric chromosome, and it involves chromosome 13, 14, 15, 21, and 22. And these individuals carry only 45 chromosomes. Here you are seeing that chromosome 21 is missing over here, and that has gone and it's sitting on chromosome number 14. So there's a 14-21 translocation. And usually, again, sometimes in these cases, there would be babies who are born with either a Down-Patau or Edwards syndrome, Down or Patau syndrome in this case. Here in this case, there is a risk for Down syndrome as well as in the family. So remember, Robertsonian translocation, the chromosome number in that individual will be 45. Now, do you do a karyotype in cases where there have been previously healthy children? That's another question that sometimes comes to our mind. Now, see this case, there's a healthy female, there were 2 miscarriages, and the 4th miscarriage, there was a 11q microdeletion which was identified. So couple karyotype was done even at that state, despite there being a healthy female, and the male here showed that there was a translocation between chromosome 5 and 13, and there was an insertion related variant here which between chromosome 11 and 12. So there was a very complex karyotype that this individual had, and we could counsel them that the outcomes of a successful conception will be less than 10%. So in this case, even if there have been cases where the previous children have been normal, again, if there are miscarriages or product of conception show abnormalities, it is indicated to consider further karyotyping. Now, there were multiple IVFs in this particular family, and a couple microarray came out to be normal. So they came with a couple microarray. So do you counsel them that their chromosomes are normal? And this is what the karyotype showed. The wife showed a balanced translocation between chromosome 8 and 13. Obviously, this is going to change the entire thing. Remember, balanced translocation responsible for pregnancy losses are not picked up on microarray. Remember this. So all patients should undergo a baseline karyotype and not a microarray. Now, again, in this case, the product of conception showed an abnormality. So there was in one of the product of conception, there was a 11p deletion. And again, there was a deletion on chromosome 15 and in the third pregnancy. There was secondary infertility and couple karyotype was reported to be normal. But you see the metaphases. I showed you some karyotypes earlier. You should be able to identify the banding pattern properly to really identify if there is a translocation or not. Poorly, chromosomes wherein the banding is not proper, you should ask to repeat a karyotype because you may miss translocation. In this particular case, the translocation was so subtle that we had to, resort to a spectral karyotype to identify that there was a translocation between chromosome 11 and 15. So very, very subtly, when you're coloring these chromosomes, you're finding that this translocation is present. Again, the outcomes for this couple is going to change. Now, obviously, polymorphic variants we come across a lot of karyotypes with these polymorphic variants, and these are usually present in more than 1% of apparently normal population. There's really doubtful association with recurrent pregnancy losses, especially for heterochromatin regions. When there are inversions, like we know inversion 9, inversion Y, they're fairly common and they're really not causal of a recurrent pregnancy loss. But nowadays, because of improved karyotype techniques, we get other inversions as well. So in some cases, we may have to resort to microarray to rule out any subtle microaberrations in these patients. Then we go to the next step, wherein a product of conception testing, which is a test of choice in this case. So usually remember that pregnancies will discontinue because of chromosomal imbalances. So balanced rearrangements are not important. So microarray can be a test of choice here on a product of conception sample. You can do a FISH or a karyotype, but you get very limited information. Microarray is going to give us more information than otherwise. Now, in this particular case, there was a trisomy 16 on a product of conception on a karyotype. Obviously, any test which is done on a product of conception should be followed by a couple karyotype. So there's trisomy 16 in the POC, couple karyotype is normal. We know that this was an accidental aneuploidy and the recurrence risk for the couple is going to be less than 1%. But in this case, there were 3 miscarriages and the product of conception microarray showed a trisomy 14. Why you should do a karyotype is because of this. The mother had a normal karyotype, but the father showed a Robertsonian translocation between 13 and 14. Obviously, because of this, the risk of, the chance of a successful conception for this couple is going to be about 10 to 15% with their own gametes. Otherwise, there could be, either deletions, duplications, which could lead to pregnancy losses. Now, whenever you find a microarray result like this, there's a gain-loss type of an abnormality. Here you can see there's a gain on chromosome 11 and a loss on chromosome 4. Gain-loss type of abnormalities always is an indication that there could be one of the partners could be a carrier of a balanced translocation. So here, if we have seen this kind of a carrier, an array result, the mother was found to be a translocation carrier between chromosomes 4 and 11. Obviously, again, the outcomes are going to change. So how do you counsel these translocation carriers? Usually, chances of successful pregnancy are 10 to 15% with own gametes. Others would end up as miscarriages, infertility-related issues, fetuses with anomalies, and rarely children may be born with physical or intellectual disabilities. Now, in very, very rare cases, as I mentioned earlier, you resort to a microarray in couples. Now, in this particular case, there was one miscarriage and there was a loss of pregnancy at 10, 11 weeks, and product of conception showed a micro deletion, a smaller deletion. It was not a complete chromosome which was involved. It was a small segment of 1.8 MB which was involved on chromosome 15q13.2. Now, in this case, we resorted to a couple microarray which was normal. Obviously, because of that, we could discuss a risk of recurrence of less than 1%, possibly because this was a de novo type of a microdeletion. So which test do we consider on a product of conception test samples? You can do a karyotype, but there is a difficulty in culture because it's a non-viable tissue. About 70-80% there could be a culture failure rate and there could be concerns regarding maternal contamination. FISH will give us information very limited to about 5 chromosomes or a few more chromosomes only. But microarray usually is an ideal test for POCs. It gives more information. It can pick up all imbalanced chromosomal rearrangement. Sometimes it can be done on a stored DNA. The patient doesn't have funds. We can store the DNA, can do it later. So generally, documenting a normal microarray and a normal karyotype will completely rule out a chromosomal cause of recurrent pregnancy losses. Hence the importance of documenting a product of conception microarray. Obviously, you can collect the samples, you can collect villi, you can collect tissue, you can collect a cord sample in a sterile container with saline gentamicin, and obviously you can do the testing. Then we go on to in a pregnancy, if you come across certain soft markers or anomalies, always remember deeper analysis is usually going to be quite helpful. So we come across multiple soft markers here. So if it's a raised NT, hypoplastic nasal bone, so which test to resolve? Obviously, the initial test can be a rapid aneuploidy screening by a FISH or QF-PCR, but a further test should preferably be a microarray because we want a more deeper analysis than only a karyotype. Obviously, in cases of where there are cost implications, you can resort to a karyotype, but remember that since we are dealing with issues which could be microdeletions and microduplications, which will not be picked up on karyotype, hence the microarray may be a preferred test. Like for example, in this case, there were 2 pregnancies. Both pregnancies had a congenital heart defect. The second pregnancy was considered for discontinuation, and that showed a 22q microdeletion. Now, when we did the parental testing thereafter, the father was a carrier of the same smaller 22q microdeletion. So this was an inherited type of a microdeletion, and the risk of recurrence in the future pregnancies was 50%. Similarly, if it's a 37-year-old, there was an intermediate risk on double marker with a mild ventriculomegaly, 10 and 11 mm. Now, if you would have done a karyotype, it would have been reported normal, but a microarray showed that there was a 211 kb deletion on chromosome 2p21. Now, here, this encompass the 6-3 gene, which causes holoprosencephaly. Obviously, the subsequent scans did show holoprosencephaly, but remember, if you would have done a karyotype, you would have missed this particular microdeletion. Microarray also gives us some more information about a condition called as uniparental disomy. So what we expect is both the chromosomes, like for example, you are seeing 2 chromosome 14, but a karyotype cannot differentiate if they are inherited from the same parent or different parents. When we did a microarray in this case, it showed a contiguous state of homozygosity on this specific chromosome 14, and it was a large one, 86.8 MB. Obviously, further studies did, by using STR markers, did confirm that this was a UPD15. So UPD means that both the chromosomes are coming from the same parent. Again, this cannot be identified by a karyotype, and you can resort to a higher resolution microarray for this. Then to the last part, wherein there is increased risk of screening test with normal scan findings. Scans is normal, but you are seeing this marker trend, which is a little abnormal, a very low PAPI, a very high beta HCG, a very low UE3. So if you're seeing this kind of an abnormal trend, obviously you are opting for an invasive testing, and you should consider further evaluation. But if it's otherwise a normal marker trend, but increased risk due to age or other factors, then NIPT may be fine in these cases. Now, a few cases here. There's a 35-year-old lady, and you can see the marker pattern is quite abnormal, adverse, but the NT is normal. So obviously the couple opted for an invasive testing because she was a high maternal age, there was a bad obstetric history, and an increased T21 risk. And this, the karyotype showed Down syndrome, and FISH and karyotype did confirm Down syndrome. But in this case, you are seeing that the NT is elevated. You're seeing there's a positive marker here, along with there's an increased risk for Edward as well as Patau syndrome because of the low markers. That is, PAPI is very low, beta HCG is low. Obviously, in this case, we did a karyotype, a microarray was done, which was normal. And obviously, we have to counsel them that it is not only about chromosomes. The raised NT and other things can be because of single gene conditions, and therefore further exome-based evaluation or genetic testing is required. So to counsel couples also is very, very crucial that even if it's a normal microarray, in presence of major markers, you may need more deeper testing. Now, this was a very unique case wherein there's a 34-year-old lady, so NT scan was normal, but you see the marker trend, the beta HCG was on the lower side and PAPI was 0.18. And that showed an increased risk for trisomy 18 and 13. Obviously, low PAPI, she was started on aspirin. The patient was not really comfortable. They did an NIPT, which was low risk, but they were not very comfortable, and they opted to consider an invasive testing, and that showed that there was a 17q microdeletion of which was about 445 kilobase pairs, and that was consistent with a condition called Pulin-de-Vries syndrome. Now, this particular condition otherwise is quite silent on USG. You're not going to find any major features, but if you look at the postnatal phenotype, there's a developmental delay, intellectual disability, seizures, and a lot of behavioral issues. So obviously, doing a deeper test in some of the cases becomes beneficial. So just a last quick recap of the investigations that we use in cytogenetics. I may take a minute. So that is one is a rapid aneuploidy testing. That is, you're doing a fish study. Where is it used? So typically you are showing a double or quadruple marker showing an increased risk, or there's some abnormality or anomaly which you're identifying. So as an adjunct to chromosomal testing for a quicker result is when you can do this. And rarely in resource limited setting, you can do a fish for as a standalone test. Karyotype is a must in all recurrent pregnancy loss and infertility cases for balanced translocation. It's a gold standard to rule out chromosomal aneuploidies. Mosaicism is only conclusively picked up on karyotype, and when a partner has a balanced translocation, then in these cases, but it requires culture, longer time for result, a lot of subjective assessments are there, and it misses microdeletion. Obviously, chromosomal microarray scores much better when there are anomalies on USG, when there are major soft markers on USG, and whenever there are in resource available settings, when a couple can afford a microarray, they should go for that. For product of conception testing, obviously it gives faster result, it is less subjective, and gives much more. So which resolution to choose? Lower resolution to screen. You're doing a testing for DMD, and as an adjunct you're utilizing a microarray, you can do a low resolution. But if there are anomalies or major soft markers, always consider a higher resolution microarray. So my take home messages would be, for recurrent pregnancy losses, IVF for implantation failures and infertility cases, couple karyotype is a must. Remember, couple karyotype. Product of conception testing, microarray may be given preference. Similarly, if there's an abnormality of a scan with major soft markers or a very abnormal marker trend pattern, prefer a microarray over a karyotype. When there's a high risk screen but no abnormal marker trend, you can do a karyotype. Obviously, you can always prenatal testing for other condition. When you're doing it for DMD, SMA, a prenatal diagnosis for such condition, you can offer the option of a karyotype or microarray to document normal chromosomes. Thank you so much for your attention.
Thank you, Dr. Datta. That was indeed fantastic talk. Very elaborate, but very succinct and precise talk on cytogenetic testing. If you don't mind, if you can stay till the end for the question.
Yes, you will have to
Join
stay,
back
Sita.
later.
Yeah. We are slightly running late on schedule. So
I'll
let me
introduce Dr. Godi. Can you please introduce Dr. Ashwin Dalal? Thank you, Dr. Datta.
Yeah. So I must first congratulate Dr. Datta while it is still fresh. It was like watching a science fiction movie. And actually, the myth that karyotype is of limited use has been properly busted by you today. And I think most gynecologists are now feeling confident that writing a karyotype is not an 18th century decision. Every couple will not require a microarray. So thank you for taking us through that journey through karyotype. And what I liked most, Sita, was that he worked backwards. He worked from the consultation desk, from the patient, and then he walked backwards. So that was what was very fetching, very, very, very useful for consultants for understanding it. So then let me just take a few seconds to thank Dr. Sunita Tendulkar, Dr. Suvarna Khadilkar, Dr. Parag Biniwale, our ICOG champion currently. Thank you, Parag, for giving us this opportunity. And Dr. Sarita Bhalerao, very dear friend. Thank you so much, Sarita. And then, of course, Sita, what a fantastic job you have done along with dear Chinmay. And you people have put together such a lovely audience. Everybody is interacting so well. So very hearty congratulations for this and wishing you all the best for the entire. And let me just get ahead and bring in Dr. Ashwin Dalal for yet another very, very microcosmic detail on this subject about single gene defects. And he is the head diagnostic division center for DNA, fingerprinting and diagnostics, Hyderabad. That is a center with a very huge volume work, as I understand that. And of course, sir is MD Pediatrics and DM in Medical Genetics, a very, very kind of rare degree to come by. And we are sure we are going to fully benefit from your deliberation on single gene defects. So here you are, and please take it forward. Thank you very much. Yes.
Thank you. Thank you for that introduction, and thank you, Chinmay, for being me. Can you see the slides? Are the slides visible?
Yes, sir.
Yeah. So very good evening to all of you, and Just now, we have heard from Dr. Chaitanya about cytogenetic testing. So if we classify the genetic disorders in humans, they broadly can be classified as the chromosomal disorders and the single gene disorders. So what Chaitanya told was applicable to the chromosomal disorders, and now what we will look at are the disorders which are single gene disorders, also called as Mendelian disorders, and how different tests again need to be used depending on the situation. So if we talk about genetic testing, it is basically testing of human DNA in its various forms. So if we test at the level of chromosomes, that is what we heard in the last class. However, these chromosomes are made up of small thread-like structures which are formed from this basic molecule of life, that is the DNA. And any uh, mutation or change in this DNA which leads to the disease can be detected using the molecular genetic test. So let us see some of the molecular genetic tests that we can employ for detecting these mutations in patients with these conditions. Now, if we talk of molecular genetic tests, they are basically based on these basic two principles, that is the complementary hybridization and polymerase chain reaction. Complementary hybridization, we all have heard about that, always guanine pairs with cytosine and adenine pairs with thymine. And that is the basis of a large number of tests which are employed, including the microarray that was discussed in the last lecture. And the second one is the polymerase chain reaction, wherein we amplify the DNA from a single copy using primers which bind and then form another product. And this way, when we do about 20, 30 times, we get millions of copies of that one particular DNA that we started. And that is the basis of almost all PCR-based tests, including the Sanger sequencing and also the next generation sequencing. So these are the two basic principles on which all tests are dependent. However, the tests are not important from the patient point of view. It is the mutation that we are looking for. And in humans, we basically have three different types of mutations. Why it is important to know this is because many of the disorders have one of these type of mutations commonly found in them. And we still do not have one test which can detect all these three types of mutations. And hence, it is important for us to know what type of mutation that we are suspecting so that we can order for an appropriate test. So if it is a point mutation, then diseases like thalassemia, cystic fibrosis, these are the diseases where point mutations are more common. Point mutations refers to small mutations like a single nucleotide change or a single nucleotide deletion or duplication. There are diseases like Huntington disease, spinocerebellar ataxia, or Fragile X syndrome, wherein there are triplet repeat expansions which lead to disease. And there are diseases like Duchenne muscular dystrophy and spinal muscular atrophy, where these are large deletions and duplications that lead to disease. So if you want to look for a point mutation, our method of choice is Sanger sequencing. If you want to look for a triplet repeat expansion, our method of choice is a short PCR or a triplet prime PCR. And if we are looking for a deletion duplication, then our method of choice remains either microarray or MLPA or real-time PCR. So it is important for us to know which test we are going to order depends on what we are expecting to find in that particular patient. So before ordering for any particular test, there are some attributes that the doctor needs to understand and some what the patient needs to understand through a pre-test counseling. The doctor basically needs to understand the principle of each test. This is very important because many of these genetic tests are not like ordering a blood sugar or a hemoglobin. Each of these tests has a different principle and also different indications. So unless a test is indicated for that particular condition, there is no point of doing a particular test. And finally, the interpretation of the result. When the result comes back, it is very important that we are able to understand the interpretation as well as the limitation of each test. This is very important to remember that none of the tests are 100% effective. And since different diseases have different mechanisms by which they cause the disease, always a test will have limitations and that need to be remembered by the doctor. Similarly, we need to explain to the patient the need and benefit of that particular test, and again the limitation of this test. So this applies to almost every test that we order in genetics. So let us begin with taking different examples of different diseases and how we apply this test. So the first common disease is Duchenne muscular dystrophy. Here we know that this is the large gene in our cells, and a large deletions as shown by these black color lines. So these are large deletions or duplications of multiple exons that are commonly seen in this condition. So since these are large deletions or duplications, and since this is the X-linked disorder where only one X chromosome is present, we can do a simple PCR reaction by designing primers for these 4 exons in this cartoon. And you can see in this patient, this particular band is absent for A. That means we can interpret that the exon A is deleted. In this patient, this and this band is absent, so we can see that B and C exons are deleted. So in a very simple way, by doing a agarose gel electrophoresis, we can look at the absence of bands and interpret that a particular exon is deleted. However, when proband is not available, or when there are duplications to be detected, this kind of testing is not useful. So this testing was done a few years back, but now we have another test that is important. Is called as multiplex ligation dependent probe amplification. You don't have to go into details of how it works, but basically we should remember that instead of the bands in agarose gel electrophoresis, because here you are using the fluorescent primers, you get these fluorescent PCR products. So you have the y-axis and the x-axis. So you can separate instead of just doing 3 exons at a time, because DMD gene has 79 exons. So to check 79 exons by agarose is very difficult. In MLPA, you can look at almost 35 exons at one go. And at the same time, because of the y-axis having quantification of fluorescence, we can even detect carriers for DMD, which cannot be detected by the agarose gel electrophoresis. So if there is a clinical situation where a child comes with lower limb weakness, difficulty in getting up, his CPK is high, we are suspecting Duchenne muscular dystrophy, we are not going to order for exome sequencing because 70% patients have deletion duplication of one or several exons. So we just order for a MLPA, look at absence of the peaks, which suggests that a particular exon is either deleted or particular set of exons is duplicated. So deletion or duplication of exons can easily be checked by doing a MLPA, a very simple and very easy technique to do. Similarly, for DMD carrier detection, if previous child is not available and we want to do carrier screening in the mother, then again we can use MLPA. And as we know, mothers, these are 2X chromosomes, so mothers will be having half the product compared to the normal, and hence this mother is carrier for 5 exon deletions in DMD for this particular group. Similarly, in a situation, a child with hypotonia, tongue fasciculation, we are suspecting spinal muscular atrophy. Again, spinal muscular atrophy is known to be caused by mutations. That's a deletion of exon 7 and 8 in the SMN1 gene. Now, very important again to remember, spinal muscular atrophy, exome sequencing is useless. You have to use MLPA to detect this deletion of the SMN1 gene exon 7, 8, which can easily detect, and the same can also be used for detecting the carriers of SMA. As we all know, the SMA carrier rate in India is almost equivalent to the thalassemia carrier rate of 2 to 3%. So all obstetricians need to keep it in mind to please recommend carrier screening for thalassemia and SMA for all your patients who come to you for planning a pregnancy. And this has already been recommended through the recent guideline released from FOXI, SFM, and Society for Indian Academy of Medical Genetics, that all couples who are planning for a pregnancy should be offered SMA and beta thalassemia carrier analysis. And this can be done by a simple MLPA assay. Also, MLPA helps us to know the SMN2 copy number, which helps in knowing the severity of disease. Similarly, in diseases like tuberous sclerosis, here again MLPA can help us to look for the deletion. So Wherever you come across a case where you want to detect deletion or duplication of one or more exons in a gene, think of MLPA. So that is important to remember. So MLPA can be used for detecting aneuploidy of chromosomes 13, 18, 21 in the prenatal sample, in chorionic villus sample, or in amniotic fluid. It can be used to detect exon level deletion duplications like in SMA and DMD. And it can also be used to look at large chromosomal deletions like Digeorge syndrome, Williams syndrome, Prader-Willi syndrome, or any other microdeletion syndrome. The next group of disorders, the single gene disorders, are the triplet repeat expansion disorders. These are disorders which are caused due to triplet repeat expansion in different parts of the gene, which include Fragile X syndrome, Huntington disease, spinocerebellar ataxias, Friedreich's ataxia, and myotonic dystrophy. So here there is expansion of this triplet repeat CAG or CTG. And a simple test like a PCR can help us to identify the presence of expansion. Here you can see there is a expansion in Huntington disease where one allele is having more number of repeats, and the same can be seen here. For higher number of repeats, we use another test called as a TP-PCR or a triplet prime PCR, especially used in conditions like Fragile X syndrome or Friedreich's ataxia. Here again, we can clearly see the expansion in the affected male compared to the normal male. So remember again, Fragile X syndrome, if you are thinking of in a child with non-syndromic intellectual disability or autism, don't order for exome sequencing. A TP-PCR will be needed to identify the mutation. Similarly, in myotonic dystrophy also, that's an autosomal dominant disorder. Again, you can see that there is expansion of repeats beyond the threshold which confirms the diagnosis. So short PCR can detect the expansions up to the limit of PCR, and TP-PCR can detect presence or absence of large expansions, but we cannot see the size of expansion, and hence a combination of these two tests should always be used. So whenever you order for a triplet repeat expansion test, make sure the lab does both these tests to give you proper results. And the exact size we cannot identify unless we do a southern blot, but exact size is usually not needed in diagnosis or prenatal diagnosis. That can be done without knowing the exact size also. Another mechanism of disease causation is DNA methylation, wherein the sequence of the DNA is normal, but what is abnormal is the methylation, and there are different conditions which are caused due to methylation abnormalities like Beckwith-Wiedemann syndrome, Russell-Silver syndrome, Prader-Willi-Engelmann syndrome. And these methylation studies are very helpful. Like if you take example of Prader-Willi syndrome, different mechanisms can lead to the same disease. It could either be a paternal deletion or a maternal uniparental disomy or an imprinting center defect. And each of these will need a different test to detect each of the mechanism. But if you do a methylation analysis, it can detect all these together. So especially when you're considering conditions like these, Beckwith-Wiedemann or Prader-Willi syndrome, think of doing a methylation analysis which can detect this abnormality. The next important type of technique that we use routinely is the Sanger sequencing. This is a most used technique for point mutations wherein we can identify the single nucleotide changes. So here the basic principle is that we take the human genomic DNA and we decide which part of the DNA needs to be sequenced. So we design primers in that part of DNA, make a PCR product, use a second primer with dideoxynucleotides which are fluorescently labeled, run it in a capillary electrophoresis, and that is when we get these nice peaks of A, T, G, C. Very easily we can interpret them using our naked eyes. And so here you can see that this is the normal person having a T at this location, and at the same location the patient is having a homozygous G. So the red peak is turning to the black peak, which suggests this is a homozygous mutation. And the parent of this patient will have a heterozygous mutation where you can see both the red and the black peaks are present. And heterozygous deletion duplication can be identified as multiple peaks here. So very easily we can identify the point mutations. However, where are we going to use Sanger sequencing? So very important when there are diseases with single mutations. So this is genetic homogeneity at the variant level. where only one or few variants are known to be responsible for majority of the patients and definitive clinical features are present. The examples being sickle cell anemia, only one mutation throughout the world in all patients. Achondroplasia, Apert syndrome, only one or two mutations in all patients. Please don't order for exome sequencing for diagnosis or prenatal diagnosis of these conditions, since only one or two mutations lead to these conditions. We can just do a Sanger sequencing. For sickle cell anemia, we have electrophoresis. For achondroplasia, the X-rays, and Apert syndrome, the clinical features. And a simple Sanger sequencing, a very cheap method, can be used to detect the mutation in diagnosis or prenatal diagnosis of this condition. Another use of Sanger sequencing is when there is a small gene. So when there are small genes with less number of exons and a definitive functional assay is available, like beta thalassemia, there are only 3 exons, there is increased SBF, and all patients of beta thalassemia have mutations only in this gene. Again, for such conditions, we should not be ordering a exome sequencing. Targeted analysis for small gene can be done like beta thalassemia, only 3 exons. We design primers, we identify the mutation, the family comes in next pregnancy, we do the prenatal diagnosis, again use Sanger sequencing, identify the presence of mutation, the patients can opt for termination of pregnancy. So these are simple scenarios where very common scenarios, no need of doing a exome sequencing. A simple Sanger sequencing can help us to know the answer. However, Sanger sequencing, as with any other test, has its own limitations. If there is a patient where you don't know the disease, or where large number of exons are there, or where multiple genes lead to the same phenotype, that is when we use another technique, that is the next generation sequencing. This is called as massively parallel sequencing, where again you start with the human DNA, but instead of looking at one part of the DNA, we just chop it into small pieces, adapter ligate it, do a few PCR reactions, and basically then just sequence all the millions of fragments that are present. And then the bioinformatic analysis helps us to identify the mutation in the patient. So next generation sequencing also has its own problems. There are millions of sequencing reads, and finding a variant is many times like finding needle in a haystack. And compared to the Sanger sequencing, which is very nicely human readable, the NGS data is not at all human readable. You need a lot of bioinformatics. And this finding a variant is very difficult many times, and that is why you end up getting a normal result many times in spite of doing the exome sequencing. So next generation sequencing in clinic, you could use either as a targeted sequencing or a exome sequencing or a whole genome sequencing, depending on what you are looking for. Targeted sequencing refers to a group of genes, like genes causing for a particular phenotype, cardiomyopathy, deafness, or blindness. There will be different panels available. And so these panels, for example, here this family had previous to MTPs for a CNS malformation. Present pregnancy showed fetal MRI showed lissencephaly and cerebellar hypoplasia. And the fetal autopsy also confirmed it. So fetal blood was sent for a lissencephaly panel because now we knew that there is lissencephaly, and lissencephaly is known to be caused due to few of these genes. And here we could identify a homozygous mutation in the relin gene, which can now be used for doing prenatal diagnosis in next pregnancy or family screening. So panels are helpful if you want to look for few genes at a time. However, exome is the most common test that we order in the clinic because you can look for mutations in all genes at a time. So when you do not know what is the exact phenotype in your fetus or in the patient, then you can use a exome sequencing. For example, here, this is a 6-year-old child who came to us with overweight, neonatal jaundice, hepatomegaly, renin-megaly, and overgrowth syndrome. Why we are seeing this case is because we thought of Beckwith-Wiedemann syndrome here. And if you look at Beckwith-Wiedemann syndrome, one disease can be caused due to so many different mechanisms. So this is the complexity of genetics that we need to keep in mind. And that exome sequencing is not the answer for all the things, because all these type of mutations will not be detected on exome sequencing. So Beckwith-Wiedemann syndrome can be caused by a chromosomal translocation, so a karyotype is needed to be done. Beckwith-Wiedemann syndrome can be caused due to microdeletion at IC1 locus, so an array CGH needs to be done. It can be caused by methylation abnormality at the IC1 locus, so a methylation analysis needed to be done. And then CDKN1C point mutations also lead to it, so we did a Sanger there. And when everything was negative, we did a exome sequencing. And then you can see here in every exome, you have about 27,000 variants, and the lab has to filter them all to reach to this particular variant. So reaching from one variant from 26,000 variant always does not happen. And that is why it's very important that you provide a lot of clinical information to the lab so that they are able to do a proper analysis of the data to get you the results. Here we could identify a single base deletion in the patient on the X chromosome. So this was GPC3 gene, which was confirmed again by Sanger. You can see here the normal person has 2 T's and the patient has only 1 T here. It is Simpson-Golodji-Wehrmann syndrome, a very similar phenotype to Beckwith-Wehrmann syndrome, where we found the mutation. So exome sequencing has really shifted the paradigm and a lot of diagnostic yield. However, we have to use it judiciously and always will not get an answer. Like in this particular patient, we had so many phenotypes of skeletal dysplasia, eye abnormality, hand abnormality. However, all tests of karyotype, microarray, exome, everything came negative because a number of reasons can lead to a negative exome, like if the coverage is not full or if there is a mutation in the non-coding region. So for a number of reasons, or the mechanism is like a structural genomic abnormality. All these reasons, you may get a negative exome sequencing. And the next option could be a genome sequencing, wherein you sequence the whole genome. The advantage of genome sequencing is the uniform coverage of non-coding regions, and the structural variant detection. So possibly it will be a one-step sequencing modality for all types of variations, but as of now, genome sequencing is not yet reached there. So please don't order for genome sequencing regularly, since doing genome sequencing after exome sequencing, the yield will increase only by about 10%. So it's not going to be a major increase in the yield. And so as of now, exome sequencing remains the standard where we are suspecting a point mutation in any of the gene, whether in the fetus or in the patient. However, we are looking for a lot of many things, new things to come up, like long read sequencing, transcriptomics, and methylome profiles. These are all going to slowly come into clinical practice, and all clinicians who order this test have to keep them well versed about these newer technologies so that you don't get carried away by what is told to you by the company people. Make yourself aware of advantage of each of the technology and how it can be applied to your patient. So in summary, MLPA is a very helpful test to detect deletion duplication. Triplet repeat expansion can be detected by PCR and a triplet prime PCR. Imprinting disorders can be detected by DNA methylation analysis. And Sanger sequencing is useful for targeted mutation analysis for single gene or where few mutations are responsible for disease. Exome genome sequencing, although they have revolutionized disease diagnosis, it is important for all clinicians to understand the principal applications and limitations of this test. And so every patient does not need a test based on next generation sequencing like exome sequencing. We need to remember which test to order in a particular situation so that we help the patient in the best possible way. So I would like to tell you about the Indian Academy of Medical Genetics, this website, and there is a fellowship available for obstetricians to do a prenatal genetics fellowship of 3 months. And this collection of 57 lectures called as Gene Top on our website, again freely available. Please make use of this facility if you are interested to know more about medical genetics. And thank you. I hope I have been able to convey to you the utility of single gene tests for diagnostics of genetic disorder.
Yeah, that was fantastic. Sita, can I have one or two comments here?
Yeah, please.
So I just wanted to know 2-3 things from Dr. Ashwin Dalal. First, before I forget, the information about the fellowship if you can be so kind as to post it into the WhatsApp group that has been created or into the chat box.
Chat
It will
box.
be, yeah, thank you so much. And I just wanted to know 2-3 things like, are these tests easily accessible to our consultants? One. Secondly, what is the cost? And the clinical application you have told in great detail. So the third thing is, what is the turnaround time? I mean, how it and or what kind of sample collection? That is the so a little bit, 1 or 2 sentences on that. Thank you.
Yeah. So many of these tests are available both from the government centers as well as the private laboratories who would be doing. What I feel is the clinicians should be aware of which test to order in a particular situation. The tests mostly would be available through different government centers. In our center, since ours is a government center, in our center the cost would be something different, and so for us, a MLPA will cost about 3,000 rupees. A Sanger sequencing based test will cost about 3,000 rupees. A TP-PCR test will cost about 2,500. So that's the range. Whereas the exome sequencing, all those would cost in different labs, in private labs, differently, like maybe about 15,000 or 18,000, depending on which lab they are approaching. The main aim of telling to all about this thing is that please don't order exome in every case. That is what is important to note. And at the same time, while ordering fetal exome sequencing, please take a lot of care to explain to the patient and talk to the lab to not report a VOUS in a fetal exome sequencing. That is probably many of the obstetricians are going to face this problem if you end up ordering a fetal exome sequencing. So we should not create more problems to the patients by ordering tests which we are not able to interpret and give them an answer, especially in clinical scenario. So I think all clinicians should know about what they are ordering and to be able to interpret and help the family.
Thank you, sir. Thank you so much for that
Thank
in-depth. Thank you. Thank you. We move on to the next.
So for the last session of today, I would like to invite the chairpersons of this session, Dr. Reema Bhatt ma'am, Professor and Head of Fetal Medicine and Perinatology, Amrita Hospital, Faridabad, and Dr. Vinita Singh ma'am, the Professor at OBGY Department, Nalanda Medical College and Hospital, and Chief Consultant, Kare Nursing Home, Patna. Vinita ma'am and Reema ma'am, over to you.
Shall I start?
Yes, please. Yeah, we can.
Yeah, so very good evening to all of you. At the very outset, I would like to thank our dynamic and very visionary President, Dr. Sunita Tandurwalkar, and very hardworking Chairperson ICOG Foxy, Dr. Parag Biniwale, Secretary General Dr. Suvarna Khadilkar, and Dr. Sarita Bhalerao, who is Secretary of ICOG Foxy. And very special thanks to my dear friend, Dr. Sita, for giving me opportunity to be part of this so informative program. The first talk will be given by Dr. Mandakini Pradhan. She is a very good friend of mine, and I've heard several times how brilliantly she talks. Today, I would like to tell her about that she was chairperson of Foxy Genetic and Fetal Medicine Committee 2019 to 2022, and at present she is professor and head of Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow, Uttar Pradesh. So over to Dr. Mandakini Pradhan. She is going to give a talk on hemoglobinopathies, which is very important in our day-to-day practice. Over to Dr. Mandakini Pradhan.
Is Madam there?
No, ma'am. Madam has not joined yet.
Huh? Not joined? Okay, I'll just call her. Then Reema, can you just introduce Ratna Madam, and then we will. Hello?
But I saw her, Sita.
Yeah, even I thought.
I think she might have just got disconnected. We'll just try to connect to her.
Yeah, 1 minute.
Ratna ma'am is here. Ratna ma'am, will you be able to take your talk first, please? You're muted, ma'am. You're muted.
No, madam, is it okay? We take your talk first. Yeah.
Yeah, sorry, I was not able to unmute myself.
Yes, ma'am. Yes, ma'am.
Yes, yes, I can take my talk, not a problem.
Dr.
Thank you
Reema,
so much.
kindly introduce madam, please. Dr. Reema?
I think Reema is also not here. I don't think Ratna ma'am needs any
But
specific
you can do the honors there.
introduction, because Ratna ma'am is one of those, stalwarts in genetics who has introduced genetics to most of us as obstetricians. I remember attending the ICMR genetics course in SGPGI way back, I think in 2004 or 5. I don't remember when, ma'am, but that was the time you were speaking on lysosomal storage disorders, and from that time we, I mean, followed up all the diseases in terms of how we should look at it in terms of genetics. Dr. Ratnapuri is the professor in genetics and chairperson of the the Institute of Medical Genetics and Genomics in Sir Gangaram Hospital, which is one of the ACE genetics institutes in the country. She's a go-to person for all the clinical queries that you have in genetics. So she has very brilliant academic career and received the IC Verma Outstanding Researcher Award in 2019, and has worked in a lot of academic bodies, including a panel of experts of WHO Collaborative Global Network for Rare Diseases and Rare Diseases International. Ma'am, your CV is extremely illustrative and very inspiring to all of us, with all your awards and all your accomplishments. I salute the way you bring the topic into a very simple format and present it in a way that we can understand. So thank you for taking this talk ahead of your schedule, and the stage is all yours.
Thank you so much for that very kind introduction, Chinmay. I have also learned along the way, and I'm able to share what I know and what I love. Can you see my screen?
Yes, ma'am.
Yeah, it's in the presenter view, no? I'll
Presenter
put
view,
it.
yes, ma'am.
Yeah, 1 minute. Sorry. Let's put it. Is this okay?
Perfect.
Yeah, okay. So I've heard both the talks by Dr. Ashwin as well as Dr. Chaitanya, and brilliantly delivered as always. And that has been an amazing introduction to what I was asked to speak upon, and that was the clinical applications of genetic testing. Now, ordering a genetic test, even if you remember what Dr. Ashwin said, is not like how we order all the tests, just, a list of tests that are written on the consultation sheet, like a CBC, a TLC, a DLC, because there is a complexity involved. Because the whole of genetics is like the book of our life. And I'm sure that just with one pen stroke, we should not write it off. And here you can see that every page is a chromosome. And on that page are words that are written, and these are the genes. And the alphabets of that word are your DNA, okay, which is the ATGC. So keeping this in the background, it's very important to understand that the kind of genetic test we order depends upon the clinical scenario. So it's like going from the bedside to the bench, and that's the way we are best rewarded with the results. Unfortunately, we don't have a one-stop solution for all clinical scenarios. Like in OB-GYN or fetal medicine, there is no one-stop solution. And because one jacket does not fit it all, we have to understand what the test is, what is our question, therefore order the test to answer that specific question. The tests have really evolved and it has revolutionized our practice. And therefore, we now know that Dr. Chaitanya had said we can look at all the chromosomes in one breath. We can look not only the 20,000 genes, but beyond. But through these cases that I'm going to show you, I'm going to tell you not only what is the appropriate test to order, but how do you interpret that test? And so all of this that is present on the slide has already been delivered, so I'm not going to spend any time on this. But what I really want to talk to everyone is about the importance of pre-test and post-test counseling. And I've highlighted the points that you have to really think about when you're ordering these tests. That first you have to understand that are we doing a screening test or are we doing a diagnostic test? We must understand the training is not a degree. The training is you must understand what you're doing. There is no test that can detect all diseases. Interpretations of the test can change over time. And we have seen that. That's something that we considered not to be significant, especially this holds true for microarray, as over a period of time, as more data has collected, has become significant. Or a change in the interpretation of a WOOS variant, 90% of them are benign. And therefore, we keep saying in prenatal testing, a WOOS variant should never be reported. Interpretations can change. You can find unexpected finding in the test that we order. And before you order everything or everything is given in a report, we have to first speak to the patient and understand, are they willing to accept uncertain information? Because all this causes a lot of distress. And pregnancies now, instead of becoming really happy affairs, have kind of like become stressful situations because of these complex tests that we are ordering where they may not be indicated. We also get results that can have implications for other family members. We have to talk about that to the families, the advantages of the test, the disadvantages of the test, and the turnaround time. And the post-test counseling, which is when we sit up with the patient and discuss the report. Again, what is the test that has been ordered? What is the relevance of the report that has come? And through some of the cases, I'm going to discuss that with you. What is a WOOS variant? In some situations, we require further testing. Also, that variants can be reclassified over a period of time because we are only learning about the human body, which is really, really very, very complex. So all these issues are to be covered in pre-test and post-test counseling. Now, just to introduce the fact and the differences between screening and diagnostic tests, you have an entire population, and these are tests that are done to a whole population to screen out a smaller screen-positive population. Okay, now these are some of the names of the screening tests that have already been alluded to. Screening for Down syndrome, ultrasound to screen for structural abnormalities, and screening for beta thalassemia and spinal muscular atrophy, both common disorders in our population. These are the tests that are done to address these screenings. Now, a screening test is not an expensive test. It can be offered to a large population. It does not give us a definite answer, but it does select a smaller group of people who are the positive population to further undergo a diagnostic test to finally find a population who has a confirmed answer. The diagnostic tests are expensive tests. They are specialized, but their advantage is that they give us a firm yes or a firm no. So the aim of the screening test is where we want to translate all the knowledge that we have in genetics and genomics for public health gains because they are applied to a population at large. We want to inform women and couples of the risk of genetic disorders and birth defects in their fetus during a future or the ongoing pregnancy in an attempt to either prevent the birth defect or identify them early for treatment. Now, when we come to the various kinds of tests in genetics, I think I took this scenario because this is something which is very cheap and it comes easily. It does not cost money, and that is a pedigree. And these are all pedigrees that have come out of clinical practice, so they exist. We first ask for a positive family history. We have to draw the pedigrees like they are drawn here. And these are 3 scenarios, and I'm going to take each one of them and say that what are we going to analyze in these pedigrees, and based on that, order the test. This is the first scenario. 3 pregnancies, The baby was born at 34 weeks but was IUD. There was polyhydramnios. Again, polyhydramnios, preterm delivery, died at 2 hours of life. This was a term appropriate for date, but was hypotonic at birth. There were no reflexes. The electrolytes and all were done. The lactate was very high. And because of this lactate, this family was referred to evaluate for an inborn error of metabolism. But you have to see the full scenario. What is a common thread between all these? That there is early neonatal death and there is polyhydramnios. And here, because the child was born hypotonic, the child was in shock, the lactate was high there. It was really not because of an inborn error of metabolism. You go back and ask the family, like you always do whenever there is polyhydramnios, that what were the fetal movements like? The third information you get from this pedigree that this is an autosomal recessive disorder because there are affected males and females in this pregnancy. And so, if you've experienced this diagnosis, a clinical diagnosis which is confirmed once, you will know it for all your patients. This is something which is very consistent with any muscle disease because movements are less, swallowing is poor. Look at this, no sucking and swallowing. So I think every word that is written on the pedigree has a meaning, only if we care to analyze it. Because once we know that we could be dealing with myotonic dystrophy, you do a shake hand with the mother because we know when it's impacted in fetal life, it is transmitted from the mother because the repeats increase much more for myotonic dystrophy when it's transmitted from the mother. And if she's unable to open her hand and relax her muscles, we know that we are dealing with myotonic dystrophy. And the test that we need to order is the TP-PCR. I'll come to that later. But these are the information that we have got. Now, whilst I initially told you that this is an autosomal recessive pedigree, but if we confirm by TP-PCR that this is myotonic dystrophy, which is a triplet repeat disorder, it is an autosomal dominant. But because the mother did not have any overt symptoms, because in this disease, the severity increases down generations. So it may look like a pseudo-recessive disorder. But we know that once we confirm by TP-PCR that the mother has repeats that are within the range to cause disease in the baby, there is a 50% chance of recurrence in each pregnancy. Prenatal diagnosis, you know what to test. Additionally, because you have diagnosed a disease in the mother, which is multisystemic, you will do all her screening of her heart, blood sugar, eyes, because these are all impacted in myotonic dystrophy. So it's not only about reproductive counseling, but it's also care of the lady who has the disease. Coming to the second pedigree. What is apparent here? Multiple affected males with autism spectrum disorder. All these males are linked through carrier females, okay? Except for this male. But if you see that and this was the lady who came with this history because she recognized that a lot of her maternal aunts had children who had autism spectrum. The second thing that you see in this pedigree is her own niece has beta thalassemia major. There is no consanguinity here. So while there is an X-linked pedigree with affected male, linked through females, commonest cause of autism is Fragile X. We will test for Fragile X by TP-PCR. Again, not picked up by exome sequencing as Dr. Ashwin has alluded to. Once we confirm that this is Fragile X by testing any one affected member, we will check whether this lady is a carrier of Fragile X or not. We will not certainly go directly and do prenatal diagnosis. There's a 50% chance that she could be a carrier, a 50% chance that she's not a carrier, and therefore she will be tested, and only after that, if she's a carrier, is prenatal testing discussed. Additionally, we also know that we have to check whether she's a carrier of beta thalassemia. because there is a 50% chance that she's a carrier of that as well. So I've barely done any tests. I've done targeted tests that are based on the pedigree. So this is the triplet primed PCR, which analyzes for Fragile X, the CGG, and it's very similar for myotonic repeats. So this is a normal male, and this is a affected male. Then this is the third scenario. Now, if you look at this pedigree, this family was actually referred because in the level 2 scan, all the limbs were short. The first child had died at 3 hours of birth. Term baby, cry was feeble, had died during transfer to a tertiary care center, had also been diagnosed to have short limbs in later gestation. So therefore, when this started happening, they were worried. But because this pedigree had not been taken, nobody realized that there was a family history of short stature and delayed dictation. This is an autosomal dominant pedigree. With this history, and looking at that, the father, because he had a very typical gestalt of a disorder that is called cleidocranial dysplasia, where the clavicles are absent or they are short. And this child during pregnancy also had absent clavicles. The first baby would have had the same disorder. And because the protective cranium is not well formed, the death could have been during transfer of this baby to the referral hospital because of an intracranial bleed. And so after counseling all this, the family wanted to continue the baby because their own outcome was good. They were intellectually normal, they were a little short, they didn't have any problems. So they continued the baby. The only precaution they had to take was to deliver the baby wherever there was a good nursery in case the baby required any care, and we did not want to transfer the baby because of the lack of a protective cranium. Now, this scenario, you can see that there are 2 pregnancies that were terminated for multiple malformations, and the product of conception karyotype was done, and this was normal. Now she came again in pregnancy, the third pregnancy where the posterior fossa was abnormal. Now, because this karyotype had been done in a POC, the couple karyotype was done during this pregnancy, the third pregnancy, and the report that came, this was a report that came to us in the clinic, was that there is a deletion of chromosome 12 in the mother and 9QH plus, and this 9QH plus has no clinical relevance. But it's important to understand that if there is such a big chromosomal deletion, how can the mother be normal? So obviously there is some misreporting here. We did not have anything to go by here because here the POC karyotype had been normal. We don't know how it was reported normal. When we repeated the karyotype in our lab, you can see that what there actually was a translocation between chromosome 18, which had been missed because the resolution of the chromosomes was very poor, and chromosome 12. And possibly because she was a carrier of a Robertsonian translocation, all 3 pregnancies would have been affected because of a chromosomal imbalance between chromosome 12 or chromosome 18, which had not been picked up in the POC karyotype that was done. Maybe it was beyond the resolution of a karyotype. So we know that whenever there are malformations like this, a more robust test is a chromosomal microarray test. So really the way forward, the best thing at the bedside is to take details of the family history, make a differential diagnosis based on the history examination and the available test results, and then apply that appropriate genetic test, which you can see in all these cases has been different, to come to a confirmed diagnosis, pre-test counseling and post-test counseling, as I've already alluded to. Now let's come to another scenario where there was this pregnancy with an increased nuchal translucency of 4 millimeters. A chorionic villus sample was done. You can either do a FISH or a QF-PCR. And actually a QF-PCR is better because you do the maternal cell contamination. and you do a microarray. Now here the microarray identified a small 1.4 million base pairs. So it's the 1.4 million ATGCs, duplication on chromosome 7q, which is the long part. You can see the dot here. It cannot be picked up on karyotype because this is beyond the resolution of a karyotype. The microarray enlarges this region and shows that this region is double. But this is not enough. You have to go into the details. You have to see what are the genes that are present in this region. And does duplication of the genes cause an abnormality or a disease? Okay. So there are sites and databases that help us to look into this. There is Decipher, there is ClinGen, or there is OMIM if it's a reported deletion duplication syndrome. So you have to look at these breakpoints. So how do you interpret a microarray report? First, you have to see what is the reference they are referring to. Is it GRCH 37 or these days it's 38? Is it a deletion or a duplication? On what chromosome it is? What is the size of the deletion or the duplication? How many genes are there in it that have been lost or gained? Does it correlate with the phenotype? And that's very important. And therefore, detailed phenotype with a detailed ultrasound, pedigree, history of previous affected is very important. And you go back to your databases and literature to review whether whether that deletion or duplication is significant or not. And we know that compared to a karyotype, the sensitivity and the specificity of a chromosomal microarray is better. And I'll come to what are the indications of doing it. Now, you can see here, actually, this is slightly incorrect. This should have been at the brother. Even though there is one not affected child, the pedigree itself alerts you to something is happening here because you have two members in the family who have recurrent miscarriages. On this side, there is one member. So actually, before this pregnancy, the karyotype should have been done with the family history. However, there was the nuchal translucency was okay. The triple screen was positive. You can see here. Repeat quadruple had been done. So therefore that was normal. So nothing was done until later in pregnancy, there was fetal growth restriction with a normal doctor. So in that fetus, when the chromosomal microarray was done, there was a double segment imbalance, a large deletion on chromosome 4 and a large duplication on chromosome 10. And whenever you have this double segment imbalance, one of the parents is a translocation carrier. And remember what I said, that this history also alludes to that. And the father was a balanced translocation carrier of a translocation between chromosome 4 and chromosome 10. Now, always, everything that comes in a chromosomal microarray is not equivalent to needing to discontinue the pregnancy. And this is one example. Here, there was ARSA on the ultrasound for which a microarray had been done and the couple had come to us for consultation with the report. The fetal microarray had reported that there was 109 kb deletion on chromosome 16. So you had to go into the details of this region. In this region, there was only one gene that was deleted, which was the OTOA gene that causes non-syndromic deafness. It's an autosomal recessive disorder. The microarray does not tell you whether the second copy of this gene is functional or not. Okay, the microarray only tells you that one copy of this gene is functional. So now what are you going to do? At 26 weeks, are you going to go and do an exome to look at this gene? What we did was we went into further details, and this was the counseling. This is all from the patient's counseling sheet that we said, we don't know the status of the second report. We know that in literature, a single gene deletion is not going to cause any disease. or even if both copies of the report are of the gene are affected, there is no severe genetic disorder that causes high morbidity or mortality. Hello? Am I heard?
Yes, madam, no
Yes,
problem.
ma'am.
Okay,
No
okay.
problem.
That causes high morbidity or mortality. But what is important to do is that please ensure that her hearing evaluation of the baby is done after birth, because we don't have this as a part of newborn screening everywhere in our country. And hearing loss is a treatable disorder. So I think more and more we have to start thinking like this, that everything that is coming up in our reports is not warranting termination of unaffected babies or those who are affected with mild disorders that have treatment. So microarray is recommended where the fetus has one or more structural abnormalities and who are undergoing prenatal testing, or even a structurally normal fetus undergoing invasive testing for a positive screen, or in IUFD stillbirth when the cytogenetic testing is indicated. It should not be offered without genetic abnormality. Every abnormality on an array may not be pathogenic, and Wu's variants must be interpreted with a lot of care. Then when I come to single gene disorders, there are two situations for beta thalassemia. One, where there is no family history, and the two, when there is a family history. Whenever there is no family history, one partner is a carrier, we must test the other partner. And if both partners are carriers, their mutations will first be identified by Sanger sequencing. And when their mutation is identified, only then is targeted testing done for the fetus. Now, in this situation, we have a family history. We check what the mutation is. All this job has already been done. We know that this lady is a carrier by HPLC, so we will only test for this mutation. Okay, she won't have any other mutation because this is what her brother has, this is what she has, and possibly what one of her parents has. On the other side, when her husband, who's also a carrier by HPLC, we'll have to do Sanger sequencing of the entire gene to identify what his mutation is. And once this is identified, only then is prenatal diagnosis done for this fetus to confirm whether the fetus is affected or not affected. We will not be doing, you know what to target in the fetus, and that is very, very important for correct prenatal testing. These are the situations where prenatal diagnosis is required, whilst these are some situations where prenatal diagnosis is not required because these do not cause any severe disease. And it's important to note this. Spinal muscular atrophy or any other autosomal recessive disorder, testing has to be done in every pregnancy because this pregnancy does not know what happened here. So the 25-75 affected versus not affected is true for every pregnancy without memory of what happened in the first pregnancy. It does not mean if the first three fetuses, if the first fetus is affected, the next three will not be affected. No, it has to be done. The risk is there for each conception. Okay. So unfortunately for her, whilst the family, the couple knew that they needed prenatal, they were guided that this is not normal because your previous babies have been affected. So a prenatal was not done and the fetus was affected. And this is how we work it out. Both parents are carriers. So if both the non-affected genes come, there is no disease. But if both affected comes, there is disease. And this is the carrier status. So carrier status is equal to normal, and at least we don't reveal that, or we reveal that, but the pregnancies are continued. Now you have to remember that there can be situations where, like I said, Wu's variants must never be reported on prenatal testing, and I'll explain to you why. So this was a family where they had two affected individuals, and you can see on the ultrasound that there was cerebellar hypoplasia, absent cavum septum, agenesis of the corpus callosum, and possibly a little bit of arthrogryposis. First pregnancy tested, nothing identified. Second pregnancy tested, really nothing was confirmed except for this WOOS variant that was reported in a gene which causes an autosomal recessive disorder, and 2 WOOS variants were reported. And so anyone could be very inclined to say, well, this is the disease and we can do prenatal diagnosis in the next pregnancy. But you have to go back and see, what does this disease cause and what does your patient have? Craniofacial features, ocular abnormality, agenesis of the corpus callosum, congenital diaphragmatic hernia. Okay, now whilst there was agenesis of the corpus callosum, the other features were not there, but I do agree that it is not important that all features be present in that or be actually can be seen on the ultrasound at 20 weeks or even at a autopsy. But what I really want to bring to everybody's attention is why it is dangerous to do prenatal diagnosis for WOS variants. Because for this very family, 3 years down the line, after the second fetus was discontinued, a gene with a new phenotype, this was a known gene which causes a brain phenotype, but it was now correlated with pontocerebellar hypoplasia. And then we went back into our database and identified so many other families, 9 affected fetuses with the COSY-related pontocerebellar hypoplasia. So you can imagine that if a prenatal had been done for that other gene WOS variant, we all would have been in deep trouble. So therefore, you have to inform the families. Well, that is what the literature is. We can only follow up on ultrasound, and a prenatal diagnosis without absolute correlation or other families in the world with that same variant that has correlated with the phenotype, if that is not found, no prenatal diagnosis should be done. It is better not to do anything rather than do incorrect things. Now look at this family, multicystic dysplastic kidneys. So normally for multicystic dysplastic kidneys, we don't do any testing. But if you go into the history, it is IVF, DADC twins. Why should DADC twins have, both of them have identical phenotypes? So based on this and absence of a real significant family history, the father had some diabetes, he had a single kidney, there was a family history of diabetes here. So there was really nothing significant in the family history except for the fact that there were identical malformations in DADC twins. So we went ahead and did exome sequencing after confirming the phenotype on autopsy and confirmed that there was a mutation in a HNF1 beta gene, a heterozygous change and autosomal dominant disorder that causes renal cysts and diabetes syndrome. Present in the father, present in this grandfather. So now you can correlate everything in this family. And we know that in autosomal dominant disorders, the phenotypes can vary. So therefore, they had a significant fetal phenotype, whilst this was diabetes. And here there was no renal problem. And here there was an isolated single kidney to which we don't give any cognizance. Now, at times, we need to do additional testing to validate and verify variants. So this was a typical radial ray abnormality. And we know that there is a host of differentials for radial ray abnormality. So the chromosomes were normal. Fanconi anemia of these is one of the commoner ones because there was no cardiac defect. And you can see here that this was a consanguineous family. So we will think of autosomal recessive disorders. So as we do for radial ray defects, we do what is called a mitomycin C stress study, which is a specific study for Fanconi anemia that shows us that there are breakages in the chromosome, which is a part of the mechanism of disease of Fanconi. Now, this made us very certain that this is Fanconi anemia. Now, when the exome was done, Fanconi anemia can be caused by mutations in one of many genes, and therefore when this NGS was done, there was a WOS variant, a heterozygous variant for an autosomal recessive disorder in a FANK gene. So when we evaluated this variant, it was not a very significant variant, but it was in a Fanconi anemia gene, so we really didn't ignore it. but we said, okay, let us look at the data and try and see if we can find the second variant. Now, when we looked at the data and we also looked at other genes, we found that in another Fanconi gene, which was the FAN-C gene, that was the FANC-A gene, the fetus had a deletion. And what we do when we find these deletions, we compare it to other data. If that part of the exons are well covered in the exome sequencing and it is missing in your patient, you know that this is a true deletion. And this was the final report, that there was a homozygous deletion from exon 4 to exon 6 in the FANC-C gene, which is the actual responsible gene for Fanconi anemia in this family. However, we may not have gone this far for Fanconi if we did not have that BREAK-IT study. So just blindly doing an exome in all fetuses that are discontinued may not really give us the results that we are looking for. So it has to be a phenotype to a genotype report, and don't accept reports like that. Now, this was a similar situation. This was a pregnancy that was terminated at 19 weeks of gestation for short, long bones. The proximal and the middle segment of the bones was reduced. There was thoracic hypoplasia, and there was mild dilatation of the left atrium. There was query polydactyly. Unfortunately, a fetal autopsy had not been done. They had come with this, and so we did a couple carrier screening, also because there was an additional family history of global developmental delay, and then a sibling death at about in the first decade of life for vomiting, loose motions, and there was also some delay here. So taking all this into account, we did a carrier screening. But what we got was a variant of uncertain significance in both partners. There were two different variants, and they were in the COL11A1 gene, which caused a skeletal dysplasia called fibrochondrogenesis. So you can see that they were VUS variants. Now, even though we got a VUS variant in a skeletal phenotype, what we told the family was, we went back into the ultrasound. We were not able to correlate the specific kind of fibrochondrogenesis phenotype. We were not sure whether all the features were there because there was no radiograph. And so for this pregnancy, we told them that we will not do prenatal diagnosis based on this, but just follow up this pregnancy with serial scan. So even though while something was there, if we had been able to correlate the phenotype or had some sample for this fetus, only then would a prenatal diagnosis have been done. So every gene reported may not be responsible for the phenotype, and do not accept any report unless you are convinced. Okay, there are many steps in confirming the genetic result. So therefore, whenever a pregnancy is terminated, if it's a skeletal dysplasia, take a radiograph, take a picture of the fetus after consent of the family, and at least store some DNA so that further testing and we can go back and confirm things. So we have to avoid it. And just before I conclude, again, an exome may not answer everything. This was a family that came because their first child had severe developmental delay, cortical migration disorder. We did many, many tests. It was a diagnostic odyssey of 4 years. Unfortunately, we did not reach for this pregnancy, which showed microcephaly in the late third trimester. Because this was something that could only be identified by a genome. And finally, when through a project, we did a genome sequencing for this girl, we identified a huge 400 base pair deletion and also a smaller deletion in the TUB GCP6 gene, which can confirm. So beyond an exome also, there are tests like Dr. Ashwin had alluded to, but this requires a lot of expertise to be able to evaluate it from the lakhs of variants that come up when a genome sequencing is done. So whilst there are many technologies and many more technologies are coming up, every technology has its advantages and disadvantages, and we have diagnostic yields. It's not as if, these tests have become redundant. Every test has its own diagnostic yield, and every new test will only add so much more to the next test, and they have their own place. And so therefore, Every step is important, right? From counseling to understanding the difference between a screening and a diagnostic test. What is a family history? What we should screen for? We have to first test the affected individual and not directly do prenatal testing. And the test that is used is dependent upon the anomaly, the gestation, the family history, and the past history. And we have to synthesize the ultrasound information to enable correct use of genetic tests. And all pregnancy discontinued for major abnormalities must have a fetal autopsy and appropriate genetic test as well as interpretation before a final diagnosis is made.
Rima, yeah, she can't unmute. Nilima, can you unmute Dr. Rima? She's joined. Thank you, Dr. Ratna, madam, for your brilliant talk as always. Yeah,
Yes,
Rima, please carry on.
yes. Ratna ma'am, thank you so much. It's been brilliant as always. So there's so much to learn. Genetics is evolving, but we need to understand that it has limitations as far as we are concerned. We need to rely on our geneticists for the decisions we take for our patients. It's correlating the genotype with the phenotype is something that is extremely important. And with so many tests available in the armamentarium, we have to be careful because every test has its limitations, as ma'am has brought about. We need to understand the family history is important. The variations of unknown significance we need to understand. We can't be reckless in our decisions when we look at the just because a test is available, we can't order it. Thank you, Ratna ma'am, for making this complicated topic so simple for all of us, and the beautiful take-home messages that we need to understand as far as genetics is concerned. Whole exome, I have made mistakes myself and got a lot of, ma'am has been guiding me that, you've made a mistake there. You should have done better at looking at the genotype, looking at the phenotype before ordering a genotype. So we need to understand that these tests have their limitations, but yes, we need to investigate adequately, taking our clinical geneticist on board in every decision that we take for our patient. Thank you, Ratna ma'am, once again.
Thank you, madam. Thank you, madam, once again. I think Dr. Vinita is there and she has introduced Dr. Mandakini, madam, so we can go ahead with madam's talk on hemoglobinopathies.
And while madam is setting up, let's just remind everybody that there was no pre-test because there are a lot of questions coming on the pre-test link, and the post-test will be only after the lectures are complete in the evening.
There's one more talk and then we'll have the post-test. Yeah. All yours, madam. Thank you.
Should I start?
Yes,
Yes,
ma'am.
madam, we can start. Yeah.
Yeah, but then
Are you able to share your screen?
yeah, just a minute. Share.
Yeah.
Yes.
No, I don't think so. Can I? Are you seeing my
We are seeing
screen?
your
Yes, ma'am.
desktop. We need to
Desktop?
see your
Okay.
presentation. Yes.
Yeah.
Yes.
Yeah,
Done?
yeah.
Yes.
Yeah, done.
Yes,
Perfect.
madam.
So that's nice, and good evening, everyone. And I'm very sorry for that. I got stuck up. In fact, I forgot that I have a lecture, frankly and truthfully. Suddenly, I realized it's already 6. I rushed from hospital because there is no Zoom link in my hospital. So I'm sitting at home, have to go back again to the hospital. Few patients are still left over. Okay. Anyway, thank you, Dr. Ratna, for making my life simple. And this is something which will be lighter than what Dr. Ratna told all of you. So this is today I'll be talking about hemoglobinopathies. Thank you, Dr. Sita, to visualize something like this, which is need of the hour. And thank you, ICOG, for inviting me, giving this platform. Sometimes I forgot to thank you, everyone. So that's why my first slide is thank you, so that I will not forget. Anyway, every one of us in our antenatal clinic, every patient, every woman are happy to have a good news. Then they have all those photograph these days, this maternity photograph. Then they go on to have some more delivery, have a sweet photograph, and then why it's not moving? Then a packet of sweets for you, and then you get more sweets, even including gujia for Holi. So, but then after 6 months, what happens? There is a bad news for that same baby. It's a you appear for a medical negligence in a court case. You are an accused over there. What happened to all those sweets? And you need to face this. And that is because the baby has developed anemia after 6 months of age. Somebody has told it is transfusion dependent anemia. It can cause iron overload, and the baby has to have chelation therapy. Heart, liver, endocrine organs are likely to be affected with iron overload. 50% do not cross 20 years of age. And the definite the treatment is by bone marrow transplantation if a match person is available, specifically sibling. There is emotional and behavioral problem that happens to this. So let's talk about what I'm talking about is hemoglobinopathy and basically thalassemia. So let's recapitulate our old this thing. This is the whole blood has got RBCs, WBC, and plasma. So let's forget about plasma. We are not talking about this disorder. We are not talking about white blood cell. We are talking about red blood cell, or what is called RBC. And RBC, each RBC has got millions of hemoglobin molecule, and that carries this oxygen. So RBC has got millions of hemoglobin molecule. Hemoglobin consists of heme and globin. One part is heme, then the other part is globin. This globin are of two types. And heme is iron and porphyrin, and hence your iron deficiency can cause a decrease in hemoglobin. So and today we are talking about what is globin, and there is a globin chain defect. In an adult, our almost all hemoglobin are hemoglobin A, or which stands for hemoglobin A. And it consists of 2 alpha chain and 2 beta chain, and hence we call it as 2 alpha 2 and beta 2. During fetal life, the fetus has got most of the hemoglobin is alpha 2 gamma 2, and it is called hemoglobin F. There are 2 alpha chains and 2 beta chains. They combine together to form one molecule. And what is we screen for is alpha 2 delta 2, and that stands for hemoglobin A2. So with this basic fundamental of alpha 2 beta 2, then we know that if there is a defect in beta, that means the hemoglobin A production or adult hemoglobin production is less, then the person is called as a beta thalassemia. It may be a carrier or it may be a major. So what happens to this hemoglobin, which is hemoglobin F in fetal life, or most of them are alpha 2 gamma 2, they gradually decrease, and at birth and by 6 months of age, it almost disappears. Whereas hemoglobin A, which is alpha 2 beta 2, starts increasing in intrauterine life, very less amount, but after birth, it increases, and by 6 months of age, it is into the adult amount. And hence, at 6 months of age, when F is nil or F disappear, hemoglobin F disappear, which consists of alpha 2 gamma 2. If there is a defect in beta, then hemoglobin A is not well formed, and the child becomes symptomatic at 6 months of age, not at birth. And hence you should remember that doing a cord blood for hemoglobin or hemoglobin HPLC cannot identify alpha, will have more of hemoglobin F, and cannot pinpoint that this hemoglobin A is not there in the fetus, or the newborn is affected with thalassemia major or not. So if alpha is defective, then it is called alpha thalassemia. Beta is defective, it is called beta thalassemia. It is as simple as this. And in beta thalassemia, the beta globin gene that is present on chromosome 11, and a normal individual, what I have done is I have drawn 2 chromosomes of 11, chromosome 11, and the gene that is beta globin gene I have represented. The boxes represents one exome or one exon, and there are 3 exons in beta globin gene. So there are 3 exons of one beta globin gene, and other chromosome has got 3 exons. So now these 2 beta globin gene, there can be a defect here, or at another point or any other point, and it causes, because the defect is on one chromosome only, and hence this situation is called as a thalassemia trait. Whereas in thalassemia major, what will happen? There are mutation or defect in both side of this beta globin gene, or both the chromosomes of the beta globin gene, and hence it is thalassemia major. And this mutation may be same as on one and the other chromosome, or may be different as seen here. So with this basic fundamental thing, we move on to what are other hemoglobinopathies that we see. One is beta thalassemia or beta thalassemia. Other hemoglobin are structural hemoglobin variant, that is hemoglobin S, or what is called as a sickle cell disease. So in our undergraduate day, we know that at 6th position, there is one change of A to T, or GAG is replaced by GTG, and hence the representation of amino acid is different. Glutamine is replaced by valine, and it causes the RBC to become as sickle shaped if exposed to hypoxic condition. So, and this is called sickle cell disease if there are both the chromosomes are affected with sickle cell, or both the beta globin gene are affected with the same mutation. Similarly, hemoglobin E, hemoglobin D Punjab, and hemoglobin C. Hemoglobin E is seen in northeast region, and there is a mutation at 6th position, then we call this a hemoglobin S. And if it is at 26th position, then it represents hemoglobin E. And at different position, that is 121 and another different 6th mutation is causes hemoglobin D and C. What I want to tell you is that a particular mutation at a particular site causes SEDC, whereas beta thalassemia mutation can be of different types and across this gene or beta globin gene. And hence, with this basic knowledge, what you can do is, when you are seeing a couple with beta thalassemia trait, you need to know what is the mutation in that couple. And I will depict how it can be different in different couple. So the carrier frequency is different. As you see, there are different, the ethnic group that has been listed over here, and the carrier frequency ranges from 3% to 18%. So the carrier frequency depends on the ethnic group, but however, overall it is 3 to 15%. Similarly, in hemoglobin E, almost in Assam, almost 50% of them are hemoglobin E carrier, and hence you need to know that a patient who is from Northeast are likely to have hemoglobin E, and similarly hemoglobin S has got some pockets in different part of India. It ranges, and carrier frequency of thalassemia also has been reported with a different geoethnic group. We may not go into detail of it. And what happens is there is decreased production of globin chain, and hence this alpha to beta 2 ratio is altered. Beta is less, and hence alpha will be more. So excess of alpha chain, and this alpha chain gets precipitated in RBC, and these RBC are hemolyzed and causing anemia. So beta thalassemia major, as per the severity, one has got the most severe form is beta thalassemia major. Then next comes is E along with beta thalassemia. On one chromosome, it is on one gene, beta globin gene, it is hemoglobin E, and another gene, it is beta thalassemia. other mutation, it will be called as E beta thalassemia, S beta thalassemia, then D beta thalassemia, and EE. Probably EE comes before D beta thalassemia, then SS, then DD. In India, SS are less manifested, but in a particular family, the SS manifestation varies. So, and the disease burden is, this is the most common single gene disorder in India, and India is the thalassemia capital. 10% of total world incidence is in India. 10,000 children born every year in India, more common in certain communities. But let's not remember that this community only will be tested. If we can test, then it can be decreased. So the treatment cost is blood transfusion, chelation therapy, lab test, hospital stay, and finally the bone marrow transplantation. All our voluntary blood donation, 40% of them are used for thalassemia transfusion. So if we are giving birth to a thalassemia baby, and that is, we as an obstetrician are responsible for bringing a thalassemia major child to this world. That's very firmly I tell this statement. And 40% of blood donation goes into transfusion of these children. And there are side effects of it, not going into detail. That's what cost is estimated. And this cost turns out to be, total cost of treatment per year is 1,67,000. As the child, the age of the child increases, the cost also moves up. For prevention of this, Ministry of Health and Family Welfare has got guideline, and I'm not going on to detail. That was published in 2016. They have different areas have been estimated that what is the frequency and prevalence of thalassemia. They have policy, and this is, I'm not going into detail, but what they recommend is CSC, PHC, blood banking services and outreach services should be screened. This adolescent schoolgirls also should be screened, and pregnant women should be screened. And but see the tragedy. There are contributors, there are 26 of them, and no gynecologist in the list. Can this Ministry of Health guideline will be implemented by gynecologist? Like it is, they expect that it is to be for pregnant women. Pregnant women will be screened, but making a guideline without a gynecologist in the list. That's the tragedy. Antenatal care provider is responsible for giving birth to a thalassemia major child. That's very firmly, I tell you. And this is in 2017 Times of India edition has stated that thalassemia test to be must for moms to be, or all pregnant women. And there is a pending this thing in the ministry also. So screening for thalassemia prevention is always better than cure. We need to screen. How do you screen? Red cell indices, MCV will be low, or hypochromia, microcytosis will be there. There is something called Nestrov test. There is hemoglobin electrophoresis, but usually confirmatory, almost like more accurate is hemoglobin HPLC, whereby you determine the hemoglobin A2. So now here is a red cell indices picture that we usually see, but don't only see the hemoglobin, but look into MCV also. If you see this MCV, it is 50, very, very less. Next comes is naked eye single tube red cell osmotic fragility test, which stands for Nestrov. The principle is that red cells in thalassemia trait are less fragile and to hypotonic solution as compared to normal RBC. So if the RBCs are normal, or the blood is from a normal individual, this will be more fragile. So they will be fragile. So another it will be more transparent because the red cells will be lysed, and the solution will become more transparent. It's a simple test for large scale screening. This was used. So if you can see that, if you can see the line, this is a paper and a line is drawn in a this thing, two test tube. There is one is distilled water and buffered saline. The RBCs are put onto it, or the blood sample are put onto it. In both the tubes, you can see the line through this tube. So you can read this line. Here, you cannot read this line, and hence beta thalassemia cells are floating in this tube, and hence you can't see this line, and that is how it is read as beta thalassemia trait. This is how the blood this test is done. This is for our almost like theoretical this thing as of now. Next comes electrophoresis. We as a student used to see this. You can identify, okay, S is more here in this line, S is more, F is more, A is more, like this. Whereas you cannot quantify exactly what is the amount it is there. There is some elution test which can quantify, but still it is not that accurate as that of hemoglobin HPLC. Then came microcolumn chromatography. It was technically difficult, but next comes is hemoglobin HPLC. HPLC will quantitate how much F is there, how much A2 is there, and then area under the curve is calculated and is represented as percentage. So A2 more than 3.5% is usually considered as beta thalassemia trait. Here the A2 is 6.1%. So here is a diagram which shows that the test report which shows that it is thalassemia trait. If I do MCV, then my positive predictive value is 37%. That means MCV is low. Doesn't mean that all of them have beta thalassemia trait. But if MCV is normal, then I can be 99% sure that it is not thalassemia trait. So if you are unable to do a HPLC, which has got 99% both positive and negative predictive value, you can look into your MCV. If it is low, then do not jump into thalassemia trait. It could be because of iron deficiency anemia, alpha thalassemia, lead poisoning. However, if it is normal, then you will be 99% sure that it is not thalassemia trait. So MCV, MCH, Nestrop, and hemoglobin HPLC, and HPLC is most accurate amongst all these screening tests. So you do a screening test, A2 estimation more than 3.5% indicates beta thalassemia trait, electrophoresis microcolumn chromatography, and HPLC is the best. If it is more than 10%, so if you are seeing a report which is hemoglobin A2 more than 10%, then it indicates hemoglobin E, because this diagram, the area under the curve, at this point only E2 comes. So A2 and E comes at the same point. The lab may tell you that A2 is say 20%. Do not say that it is beta thalassemia trait. Read it as hemoglobin E. So any A2 more than 10%, it indicates hemoglobin E. So if it is, we know that it is an autosomal recessive condition. Parents has to be carrier, but they look normal. So your screening test will pick them up. If both of them are carrier, then there is 50% risk of the child being carrier. However, 25% risk of thalassemia major, which we are worried about, and we can do a prenatal diagnosis for that. So first prenatal visit, do a MCV, MCH. If possible, your pocket allows, then do HPLC. What we do, hemoglobin less, MCV and MCH less. With a normal A2, think about iron deficiency anemia and alpha thalassemia carrier. If it is more, then it could be beta thalassemia trait. And hemoglobin variant can be picked up by hemoglobin HPLC. So S, E, C, D, I told you A2 more than 10% is E. And then testing of the partner. If the partner is normal, then do not be very worried. Counsel, and however your pregnant woman is thalassemia trait, and hence treat her, take care of her hemoglobin. But if the partner is again thalassemia carrier, then you need to move on to prenatal testing. That the fetus is likely to have 25% risk of being affected with thalassemia major. It is up to the couple to take a decision for doing a prenatal testing and continuing the pregnancy, or without prenatal testing continuing the pregnancy. But please give them adequate information so that they can be offered a testing. And as I said, all of them, they look similar, but if you look closely, they are different. Similarly, molecular defect are different in different beta thalassemia. Like this family A has got the defect, molecular defect same. I have named them as A, and the fetus is likely to have similar. The lab will look into A and A. Whereas this family 2 has got a defect B in husband and defect A in wife, and hence the molecular defect could be different. And hence, this knowledge will help you in sending a blood sample of both husband and wife, preferably doing a mutation analysis of the couple before planning a prenatal testing. So the lab has to identify this A and B, and that same A and B are looked in fetal sample that you send in after doing a prenatal testing. There are more than 200 types of mutation that has been reported, and hence you need to do it. Both partners' mutation needs to be identified first, and what sample you will send? Blood in EDTA. And fetal sample is either chorionic villi or amniotic fluid. Lab reports, cost and time are variable. You need to explain them by counseling about disease, prognosis, treatment option, risk of the fetus being affected, prenatal procedure, test, and option of pregnancy termination, and our MTP Act. These are all ultrasound guided procedure. Blood group, if negative, then ICT needs to be done. Placenta to be identified.
Sorry
CPS
to interrupt you.
analysis, I should
Your
not go
time is up,
into.
ma'am.
Yeah?
Your time is up.
Okay. So I'm this thing. Whom to screen? Universal, pregnant or premarital, adolescent, previous history of affected child, family history of affected child, pregnant women with anemia not responding to iron therapy. So it should be universal. By screening, all these countries have decreased their thalassemia, like UK, Italy, Greece, and Cyprus, they have reduced it to 0. So we can also should achieve that. What Iran has done, when there was no this thing of termination of pregnancy. So they have introduced antenatal diagnosis in 2001. Before that, in 1998, the man were tested first, and the women were tested. If the man was supposed to marry a woman, then they were tested. If both of them have microcytosis, then they were not allowed to marry or not allowed to have limited family size. So by that process, they decrease their thalassemia incidences, and antenatal diagnosis and abortion have decreased it further. So if that can be done, prenatal screening for hemoglobinopathies is mandatory in Iran. So if that can be done, then we can stop thalassemia. So I must share this thing that this woman came primary software engineer referred to us at 13 weeks for ultrasound suggestive of cholelithiasis, which was incidental. It was in the mother somehow, and moderate pallor. Antenatal investigation, including hemoglobin HbA1c, was given. She shifted to Pune, showed her report, which was hemoglobin A2 of 6, downloaded online, and now somebody has interpreted to be normal, and her ANC reports interpreted to be normal. She revisited our OPD at 30 weeks. Naturally, she was beta thalassemia trait. Husband was tested, and unfortunately, the husband was found to be thalassemia trait. 4.5% hemoglobin A2. So now 32 weeks, counseling done, patient delivered 3 years ago, Lucknow, female baby. Outcome, thalassemia major. This is most unfortunate thing that can happen. So you can see this, if you go to pediatrics ward, then you will see something 5 babies on transfusion. 2 of them are brothers. So you can see them hanging these bags, which you might have delivered few of them. So, and without bringing notice to you, so to conclude, offer HPLC to all pregnant women. A2 more than 3.5% is beta thalassemia trait. If it is trait, then test the partner. If thalassemia trait, then test fetus. Prior identification of mutation by sending a EDTA sample to the blood. Fetus measure, then you have an option of medical termination of pregnancy. Testing of alpha thalassemia is only for non-immune hydrops. Please do not jump onto this thing. So I have, with this liberty of being this forum, I invite you all to SBS ICON 2025, which we will be conducting in Lucknow in 22nd to 25th August 2025. We have at SCPJMS Lucknow, We have got a genetics workshop from sampling to interpreting. So we'll be showing how to take a even blood sample. So leave apart fetal sample, maternal sample also. Then CTG workshop and bereavement counseling workshop. Thank you all for this hearing and giving me an opportunity to talk to you. Thank you.
Thank you. Thank you, Madam. Yeah, Dr. Rima, Dr. Vinita, if she's still there, you can give your comments.
Yeah.
Thank you, Madam, for your wonderful talk. Thank you. Yeah, Dr. Rima, can you unmute? Hello? Gauri, can you please unmute Dr. Rima? Dr. Nilima, can you unmute Dr. Rima?
Okay.
Yeah.
Thank you, Dr. Mandakini, for that elaborate talk. So I think it's criminal in today's era if we don't screen for thalassemia. We have to do HPLC for all patients and at least look at the hemoglobin report very carefully, as Dr. Mandakini talked about. We need to look at the MCV, the MCH. We need to see clues as to if this patient could probably be a thalassemia minor. And I think that's our responsibility. We have to take on that responsibility and definitely not have any baby with thalassemia major being born. Thank you so much, Dr. Mandakini. And thank you, Dr. Sita, for this wonderful event that you've organized
Thank you.
for
Thank you so
everybody.
much, Dr. Rima, for joining in. Please do not leave. We have the post-test now. Everybody's been waiting for, and then we can address the questions. So, in the chat box, the link will be given. So the link is on now, and you can please click on the link. It's a Google form which you need to fill, and you will be given 10 minutes. For that, we have 20 questions, and then the answers will be discussed. And then if there are any questions, we can address them also. So we will not answer those questions now. If you all can we start the timer now? Has everybody got the link? Or we'll wait for 2 minutes till everybody takes in the link and then start it. Or the timer is already started, right? Okay. So you can answer the questions in the Google form. It's there. And please remember to submit it in time. They're very easy questions. If you have heard the lectures, I don't think it should be. There are only 20 of them. And thank you to all the speakers to have given in their questions. We will be displaying the answers once the time is up for answering the questions. 2 minutes left. I have to scan and send from another phone. Oh dear. Thank you everyone for attempting the first day MCQs post-test. I think if you have submitted, it will come through. There is unfortunately no way to tell you right now whether it has gone through, but as with any Google form, if you've submitted, it will definitely come through. I think we'll quickly go through the answers. ICOG team, can we just put up I will just read out the answers for today, and every day the answers will be displayed at the end of the post-test. And if we have time and the faculties will be there, they can answer the questions that are there in the chat box. So the first question was, can we make it, yeah. The preferred anticoagulant for molecular genetics for mutation analysis is EDTA. Second, the test of choice for detection of mutation in a patient with beta thalassemia is Sanger sequencing. The molecular genetic test of choice to detect triplet repeat expansion in neurological disorder like Fragile X is triplet primed PCR. Most of the spinal muscular atrophy patients have deletion of exon 7 and SMN1 gene. The method of choice to detect this mutation is not Sanger or anything else. It's MLPA, multiplex ligation dependent probe amplification. Fifth is, which is the following is not a benefit of pedigree drawing, which is developing gene therapies, but of course it has benefits with the other ones. What does the following symbol represent in a pedigree? Question number 6. Can you just move down a bit? Yeah. What does the following symbol represent in a pedigree? It is pregnancy, not proband or positive for genetic mutation. Question number 7. How is consanguinity represented in a pedigree? It is 2 horizontal lines connecting the couple, right? Question number 8, identify the pattern of inheritance in the following pedigree. We can see that the parents are carriers and it is, so it is affecting, so it is autosomal recessive, that is 25% recurrence, affectance. Mode of inheritance, question number 9, and the given figure is likely to be, as you can see here, it is mitochondrial, right? It is not recessive or X-linked. It is mitochondrial here. In the question number 10, in a family with mother affected with X-linked recessive disorder, which is of the following is true? It is X-linked recessive. So 50% of the boys will be affected. Question number 11, which of the following is not true about chromosomal disorders? Not true again, the question is. So it is chromosomal aberrations decrease with advanced maternal age. Question number 12, following is true about acrocentric chromosomes except that they are not responsible for recurrent pregnancy losses, right? Question number 13, following statements about chromosomal microarray are true except that it will identify balanced reciprocal translocations. That's why we need the karyotype to confirm that. Question number 14, following is not true about cytogenetic testing for fetal anomalies. that whole exome should be the first hire for these. You should not be doing that. And it is basically okay, the other ones are not, but the main, that is the whole exome should be a first hire is not true. Question number 15. All are true about thalassemia major except child presents at birth with severe anemia. We saw the graph where madam showed us that it doesn't present at birth. Question number 16. Beta thalassemia trait is diagnosed by all except one of the following tests, that is HbF more than 90%, that is except one of the following tests. So HbF more than 90% will not diagnose beta thalassemia trait. Question number 17, following statements about thalassemia is correct except that both the parents are carriers, chance of fetus being affected is 50%. That is not right. Next, question number 18. The advantage of chromosomal microarray over a karyotype test is that you have a higher diagnostic yield than a karyotype of 15 to 20%. Question number 19. What is the recommended approach for genetic testing when a fetus presents with multiple anomalies? It's definitely not a screening test, but yes, you will need a diagnostic test, but chromosomal microanalysis followed by exome sequencing if required. And the last one is, which of the following genetic testing methods evaluates only the protein coding regions of the genome, which is the exon? So it will be an exome sequencing. So thank you all. These are the questions for. Can you please unmute Dr. Chinmay? She's not able to.
Yeah, thank you. No, that one question whether X-linked recessive mother is affected.
Yeah.
That answer should be all boys will be affected, right?
Can you just post it again?
Yeah.
Team Go. I see. Hello.
Can we have the questions again on the screen, please?
Gauri, please.
Hello, Gauri. Can you just post this again?
X-linked mother recessive
Yeah,
condition and mother
it
is
was
affected.
50%, right?
So that means that
All
mother will
mothers.
have both the genes which are affected. No, both X chromosomes will have the genes. So she will have all the boys who are affected.
Can we just put the
Yeah.
questions again, please? We just.
Shelly thinks the test was a bit tough.
Oh.
But that's what it's supposed to be.
Yes, all
Gauri,
boys.
please display the answers.
Wait, let me just get that.
Who was projecting it right now?
Gauri was
Gauri,
projecting.
technical team, yeah. Yeah.
Yeah. Just go on top. Go question number 15. No.
That X-linked recessive.
Go down, go down. No, go up, I mean. ऊपर जाओ.
Question number 1 से शुरू करो, 1 पे जाओ.
हाँ, इधर आ गया. 9. Question number 9. नहीं, 10 होगा फिर. 10 नीचे जाओ. Yeah.
A family with mother affected with an X-linked recessive condition.
Okay.
I think all boys will be
All
affected.
boys will be affected. Yes, that will be the right answer. Yes.
And any doubts anyone has in the pedigree or anything? If the mitochondrial, if you see, it's the mother who is transmitting it. Wherever the father is affected, it is not getting transmitted. So it affects both males and females, but the transmission is only through females.
Can you just go up a bit to show the mother being affected there? And yeah, that is what it shows you.
Somebody says 50% is right. Who is that? NP, Navya Pillai. 50% is right when it is the mother is a carrier of a X-linked recessive condition. But if the mother is affected with an X-linked recessive condition,
Madam,
that
Mandakini, madam, what do you think is the answer for 10?
Yeah. No, I'm muted. Asked to unmute.
No,
we can hear you. We can hear you.
Yeah, you
Question number
can
10.
hear. Yeah. So then this
नीचे जाओ 10. Question number 10 दिखाओ गौरी. हाँ, क्वेश्चन डी क्या है? और थोड़ा ऊपर जाओ। डी ऑप्शन क्या है?
I agree with what Dr. Chinmay says. There is some trick to it. That it is mother is affected with X-linked. Mother will be affected only if her both the chromosomes are abnormal. So X is abnormal, X is abnormal, both the X are abnormal and X-linked recessive condition. So both X are abnormal. She will transmit one X to the male. And one of both are abnormal. So she will transmit all males will receive either of the X which are abnormal. So all males will be affected.
Yes. Thank you, ma'am.
Yeah.
Because it is affected. It is
affected.
Yeah.
Yeah, that is the.
It would have
that is
been
the
in a
trick, yeah.
family with mother, carrier of X-linked recessive
yeah.
disorder, then it would have been 50% boys will be affected.
Okay, so this will be team. Can you just note that the answer will be A here, not C? Yeah. Okay, so they
Yes,
want
ma'am.
15 minutes. Tomorrow's will not be that was it? Okay.
No, it's not difficult. I think all the speakers had
It's
addressed.
for an obstetrician, it is little difficult. I will not like it should be 50% should be very easy, like sample collection, then doing karyotype, because people are confused with karyotype and going above like chromosomal microarray and then going into it is little difficult. This consanguinity is good. This EDTA is excellent.
Can you
Like
mute
one should
Dr.
know that.
Shobha, please?
I think, ma'am, all the speakers, they kind of addressed all the issues.
Yeah,
So
I
these
think all.
questions are totally on the lectures that they gave.
Okay.
So that way it's okay, but
I
yes,
think
in
all the
one
questions
glance.
had, yeah, absolutely. All the questions were from their PPTs. We didn't, they didn't ask anything outside their presentations. So they have covered all that.
Okay.
No, but it was an excellent evening, ma'am. Lot of learning.
Lot of
All
learning, yes.
the lectures. How many times we listen to all of you, we still learn more and more.
So there were a few questions. What if, no, something we quickly have, do we have time for that or?
Can females be affected with X-linked recessive disorder?
Yes.
Yeah, you can,
If
madam.
both the,
Dakini ma'am is here, yes.
yeah. But if both the X are abnormal, then it can be. The question says female is affected with rare condition, but yes.
Yeah, yes, possible. Question number 17, shouldn't it be 10%? What is question number 17? Oh, at least somebody found it was rather easy. Jayanta sir found it rather easy.
Question number 17, I thought.
Yeah, Navya is also having the same doubt, but I think that got sorted out.
Yeah. I think we just.
Shobha ma'am is calling, Dr. Shobha Gudi.
Yeah, can you unmute Dr. Shobha Guri, please? I've told. Gauri, can you please unmute Dr. Shobha Guri? Results will be given to me.
17 is 17 is if both partners are carrier of beta thalassemia, then chance of fetus affected with beta thalassemia major will be 25%. So it's written 50%. That is.
Option should be 10%.
No, 50%.
No,
25%.
No, okay. Yeah, exactly. Dr. Chatterjee is written if you've heard all the lectures.
That is for sure. Mandakini, I just wanted to say one thing is I think some amongst the audience were confused about autosomal recessive. They were thinking that autosomal recessive, the individual or the index person cannot manifest the disease. Sometimes they get that thought, because it's autosomal recessive, they will not manifest, they will be asymptomatic. But if they carry the gene in both the alleles, if they're homozygous for the gene, they will manifest, they will become the index case. In the X-linked recessive question,
Okay,
if they carry, they become the
this
index
is in
case.
X-linked recessive.
In
Yes.
X-linked recessive.
This question number 10 is little.
Yes. No, but I think they were confused that a woman cannot manifest. She can manifest. She
yes.
can manifest, and
Manifest
when she has,
can become,
then
can marry also.
Yes, she can have reproduction also, and then all boys will be then affected. So Chinmay and Mandakini both were right in that. So don't get confused that recessive gene cannot manifest in our women. It can manifest. They feel that recessive women will not, they will be kind of a carrier only. That is not true. They can manifest.
No.
Okay, there was one question that in cases of polymorphisms and if you get an RPL, what do we do? Madam, do you want
Polymorphism on which gene?
They have not. One patient
So
has
any talk about polymorphism? What is a polymorphism? Definition? So little more. Anyway, subsequent this thing in RPL, you need to
I
look.
think we are going to be addressing RPL on the third day, but Dr.
That must
Kapoor
be.
will be there. We will take that up because of short of time here. Dr. Sakshi has written that it is 50%, so there is a bit of discrepancy in that.
What? Which one, Sita?
The same question number 10.
10, yeah.
No.
It's little tricky.
Slightly tricky. That's why they're not understanding X-linked recessive. They should first read the disease X-linked recessive. Then. No, because see, Mandakini, I'll tell you, there is some concept that one X is necessary, no?
Yeah.
For normal life.
I know. Even
so if both are
when I was
affected, they
answering,
feel
I
that
got confused.
correct.
Can
So
you unmute Dr.
one normal
Sakshi?
X is necessary, they will think. That is why they are responding like that.
Can
But
you
this
unmute Dr.
is a small gene which is abnormal. The rest of the genes are normal. So the individual will manifest the disease. Yes.
Team, can you unmute Dr. Sakshi, please? There's a question, madam. How do you manage if both husband and wife sickle cell positive? Carrier?
Yeah, I just answered. If
Okay,
both husband and wife are sickle cell positive, means sickling positive, then there is again 25% risk of recurrence.
Same as autism.
And you do a prenatal testing after counseling. And we do prenatal testing. But if one of them is a sickle cell disease, then one sickle cell disease marries to a sickle cell trait, then the chances of normal is not there, but there will be 50% chance that the child will be sickle cell disease and 50% chance that the child will be sickle cell trait. Okay. Now, if a husband or a wife is sickle cell disease, that family, after knowing that, okay, disease is managed well with us, so they may not opt for a prenatal testing. Many couple, after explaining, they do not opt for a disease which they already are suffering with, like they are having with. So let's not call it as suffering. Let's call it as, after explaining whether they want a prenatal testing and termination of pregnancy if the fetus is diagnosed to be affected. If they are not willing for that, then probably we should not go ahead and do a prenatal testing. Or if at all they want to know if the fetus is affected or not, then you can. So a lot of counseling needs to be done. Otherwise, you can go ahead and do a prenatal testing.
Is Dr. Sakshi unmuted? Is she still there? I don't know. I think we have left. All right. Okay. Is there any other question?
There's one interesting
There is one thing, Chinmay and Mandakini, whether that X-linked recessive thing is the mother is affected. No, the mother is affected and the father is a carrier. Then all the sons will be affected?
If the mother is affected and the father is a carrier, then
father is also affected.
All the X
father has got
chromosomes.
only one X.
Yeah. So all the X chromosomes will be abnormal. So in that case, the whole combination, whatever they will have, will be
all will be.
yeah, theoretically. Because you will have an X-linked recessive condition where all the X chromosomes are abnormal in the female as well as the male. So there is no normal X chromosome that will ever get transmitted. So all males and female, that is a very theoretical
It's a
possibility.
very
But
rare combination, though.
yeah. And similarly, Navya has asked a question about what will be the survival of that person. So we are not debating on the survival per se. It's a theoretical possibility that if there is a person who is X-linked recessive and a female and carries both the abnormal genes, then the chances of transmitting that abnormal X to all the baby boys, and therefore all boys, will be affected. Survival, well, you can, because if you give factor 7 replacement and factor 8 replacement and they are taken care of properly, then survival rates will be there. I mean, it's not impossible. Also, somebody has mentioned a term called lionization, and that is another reason why an X-linked recessive disease may manifest in a female, even if she is carrying just one of the alleles. Mandakini ma'am, would you want to elaborate on that?
Why a female will manifest a X-linked disease? Say, for example, DMD, Duchenne muscular dystrophy. We all of us know that it is X-linked recessive. If a female is a carrier of X-linked recessive, she should not manifest. But here is a female who is manifesting DMD or Duchenne muscular dystrophy. What could be the causes? One is lionization, as Dr. Chinmay said, that one X is silenced. So the normal X in this female is silenced, and hence the abnormal X is manifesting or functioning. That is one condition. Second is, all of us know that it could be mosaic Turner syndrome. So Turner have got only one X chromosome. This X is abnormal. So hence that they will manifest, and hence a woman who has X-linked recessive disorder manifesting, first test to be done is a karyotype. Then next is, if it is there is a X, there is a translocation between X and another autosome, and the translocation point has gone through this DMD gene. The gene is a long gene. Now, if the translocation point will just chop off this this gene, passes through this gene. So half of the gene is in X, other half is in the translocated region. So this gene is no more functional, though there is a balanced translocation. So X autosome translocation traversing in between the gene, in the gene, is one another cause. So these are causes of X-linked recessive being manifested in the female.
I think the more we will learn about these,
correct.
more we realize how
It is a maze. There is one question, Sita, by Prasanna Kothivale, no? Prasanna Kothivale. Mother is beta thal trait and husband is HBD. I have this couple in my OPD right now. It's written.
D beta thal.
D beta.
So the fetus can have D beta thal.
D beta thal.
D beta thal are, say for example, they may manifest as thalassemia intermedia, is a clinical manifestation that is a hemoglobin between 7 to 10. So they are they are very, very mild. It could be thalassemia trait or thalassemia intermedia, not beyond that. It will not be a transfusion dependent anemia. So it needs little amount of counseling, and then if they want, they can go for testing, but usually it is not recommended as such for D. They are so mild.
One good thing they have asked is how prevalence of disease or incidence of carriers can be predicted.
Can be predicted by reading through the published literature.
No. See, what happens is, what the question is very valid. Many patients ask that. Suppose we say that it is 50% affected, 25% carrier, like that. We are always going by the arithmetic of 4 offsprings. And then we are saying that this much, right? But in practical situation, if this is A, B, C, D, when the couple conceives, we don't know whether that is A or B or C or D, right? We don't know. So it can never be said. So the couple can
counseling of the
continuously have
patient,
affected or
we
continuously have carrier or continuously have normal or a mix
so
of all this. Yeah.
for to explain the patient, it is better way that you tell them that in a box you put 25 red balls and 75 white balls. Close your eyes and pick up one ball. It may so happen that you may pick up first is red, second is red, third is white. The sequence of red, white, red doesn't hold true here. So this is how we
Absolutely.
explain the patient.
Correct. That's a
Because
very
every
nice trick.
pregnancy, that is
The
very
memory
nice
Every
loss.
example.
pregnancy has got 25% risk.
25%.
And to explain the patient, this is the analogy that I use.
Very practical, ma'am. Very nice. I think we'll
That's a good way
steal
of,
your
That's a
analogy.
very wonderful, very wonderful.
way of putting it. Yeah. Okay, since we've found these, many of them have found today's post test a bit difficult, we will make sure the other two are not so difficult, so
Yeah.
that overall you get more than 50% to pass, right? It's not based
Or
on
you
everything
reduce the
altogether.
pass. You
No,
reduce
no,
the
ma'am,
pass
no.
percent.
No, that's
Because this is so tough. No, because it is
Eh,
taking a lot of
no,
explaining,
NBE
right?
has done it, NBE has done it, and NMC has done it. So ICOG should do it. Yeah.
Yeah.
Okay,
Okay,
great.
I
No,
think
ma'am,
we'll
it was
close it
wonderful.
for
I mean,
today. Thank you so much.
entire session, Dr. Sita,
Thank
was
you.
Wonderful. Thank you so much. We'll get the answers again. Um, we will post it again tomorrow, okay? We will post the answers tomorrow because today many of them have left. So we will post the answers in the chat box tomorrow as a request. But I would like to take this opportunity to thank everyone who's been here till now. We've had a wonderful record attendance, I think, of more than almost 450 at the beginning. So I think it was very useful, very informative, and very practically oriented because a lot of us faced with these scenarios. And then, of course, we need to take the geneticists on board, but it is our responsibility to understand what we are asking for as well. So to begin with, I thank the Foxy president, Dr. Sunita Tandulwadkar, to give us this opportunity. The ICOG chairperson, Dr. Parag Biniwale, for letting us have this certification course. Dr. Sarita Bhalerao, the secretary, for making it so easy for us. Dr. Suvarna Khadilkar, the secretary general, and the other ICOG office bearers, Dr. Ashok Kumar and Dr. Sheila Mane. And a big thank you to all the chairpersons. The first chairpersons, Dr. Radhika, Dr. Meena Samant, Dr. Shobha Gudi, Dr. Reema Bhatt, and Dr. Vinita Singh. And of course, to all the speakers who've done a brilliant job. I mean, this would not have been a success if these speakers were not there. So thank you once again to Dr. Sakshi Yadav for Shagun to get the basics right in terms of pedigree charting and basics of genetics. Dr. Chaitanya Datar and Dr. Ashwin Dalal for beautifully explaining the relevance, the cytogenetic testing and the testing for single gene disorders in such a wonderful way. And of course, the bosses, Dr. Ratnapuri Madam, for giving a wonderful talk on practical applications of these tests, how they can be used and how we should be judiciously using each test and not just randomly asking for these tests, and must to interpret also them correctly with the help of the geneticists. And the last but not the least, the must screening test for thalassemia of hemoglobinopathies coming from Dr. Mandakini Madam, so beautifully explained that it is criminal in this day and age to let a baby be born. Actually, of course, it's the parents' decision, but at least we should be able to offer them that and know beforehand if the child is going to have thalassemia major. So thank you all once again. And as I said, we will have the answers put up once again tomorrow, and we will see to it that the questions are not that difficult. Thank you. Thank you, Chinmay, for being there.
Well, thank you so much, Dr. Sita.
And Dr. Kannan as well for coordinating this beautifully. And of course, the ICOG office
you,
team,
ma'am.
Dr. Lima, Gauri, everyone to do this. It is a task, I know, to get all the questions and everything, but thank you all. Thank you. So we meet again tomorrow at 4 o'clock sharp to have the session on fetal medicine. And that will be convened by Dr. Chinmay with Dr. Navya as the coordinator. Thank you. I hope I have not missed anyone in thanking. Thank you.